To start the day, we have Dr. Aman Bhalla taking us through the tutorial titled Asymmetric Synthesis in Drug Designs, Auxiliaries and Reactions. This tutorial focuses on the role of chiral auxiliaries and key reaction strategies that play a crucial role in the design of enantioselective therapeutic agents. Please join me in welcoming Dr. Aman Bhalla.
Thank you, Tanya. Okay, just a second. So very good morning to all. Okay. So I guess if I ask about the chiral auxiliary, at least you people have information or a definition about it, or you can tell me the chiral auxiliary used in which generation? Second. I am very happy. At least you start in a positive response. Okay. So we have studied about the all generations and the basic introduction methods. Okay. When we have discussed about the generations, we came across a generation where we have to use a auxiliary. Why we have to use a auxiliary? Because our starting substrate does not have any chiral unit. And why a chiral unit is required in a molecule? Because we said that a chiral environment is required to distinguish between the two phases. Okay. So if you see on screen, we are writing chiral enolate, chiral auxiliary, SAMP, RAMP, sultam. These are the common names, some with respect to the nomenclature of the molecule, some with respect to the scientists who discovered them. Okay. So if you see the picture which I have depicted here, before going to the chemical reaction and the mechanism aspect that yesterday you have pointed out regarding this, you have to know how the reaction is going to start with respect to two important models which we have to study. Now see in your screen, what is the prochiral center in your substrate? It is the carbonyl compound, carbonyl group. That carbonyl group is going to attacked by a nucleophile. I guess everybody know the objective, that you have a prochiral carbonyl group. You are going to add the nucleophile at the carbon of the prochiral carbonyl functionality. But the entry of the incoming nucleophile is always dependent on what? What is present in adjacent to carbonyl carbon. So if you see a chiral center in 3.7, you see chiral center we marked it small, medium and large group. So this is the first model which Cramps propose it, like if the group has to enter to the carbonyl carbon plane, it must come from the side of smaller group. Okay. So now I wanted that this answer should be come from your side. If you are just looking the picture, there is no bigger mechanism details that you have to study. Just see the picture. As a chemist, as a researcher, what you can interpret, that if you are bringing a nucleophile to the carbonyl carbon and that entry is guided by the presence of groups adjacent to the carbonyl carbon, तो adjacent to the carbonyl carbon आपके chiral center में कौन से ग्रुप प्रेजेंट है? तीन ही हो सकते हैं, because one carbon bond is joined with the carbonyl group. So that's mean you have to distinguish that three group which are present close to the carbonyl. How can you distinguish them? Small, medium and large. Now if you see in your final 3.8, the entry of the nucleophile is occur from the side of small. न्यूक्लियोफाइल को बेनिफिट कहां से मिलेगा? फ्रॉम द साइड वेयर अ स्मॉल ग्रुप इज प्रेजेंट. ओके, सो इट ऑलवेज डिपेंड द प्रेजेंस ऑफ द स्मॉल, मीडियम, लार्ज ग्रुप that is guiding to conversion of sp2 carbon to sp3 carbon in a way that it will lead to a very less steric hindrance. So now if I am going to ask what is the structure or geometry of methane molecule? It is tetrahedral in nature, you call it sp3. How you place all four hydrogen atom in space? आपने कभी ऐसे तो नहीं किया, ये कार्बन ड्रॉ कर रहा हूं, वन हाइड्रोजन, सेकेंड हाइड्रोजन, थर्ड हाइड्रोजन एंड फोर हाइड्रोजन. डिड वी डू दिस? वी कैन डू इट. हमारे पास तो 360 डिग्री का पूरा 3 dimensional स्पेस है. वाई वी आर प्लेसिंग इट लाइक अ 4 corners which have an angle of 109? So that if nature says I am giving you 360 degree angle, try to place the group at maximum distance apart so that there will be no steric crowding. Okay, so in that way if you have a small, medium, large group, it is always preferable that the incoming group should comes towards the side of small group so that there will be very less steric hindrance. So the energy of your transition state and intermediate should be low, because if steric hindrance strain will be less, you will feel easily. Okay. So that is the Cram's model, but then comes the Falcon model. It just further elaborated it. What it elaborated? It is saying that if you are going to make any sp3 molecule, sp3 has a tetrahedral geometry, I guess everybody know. So the incoming group always prefer to make an angle of 109 degree so that they can properly fix in the tetrahedral geometry. Okay. So whenever I am going to say that perform an asymmetric reaction on any carbonyl substrate, what comes to your mind immediately? That whenever you are converting your sp2 carbon to sp3, then the open chain model has to be followed. Okay. So this is the one criteria which we have to see in mechanism, that how the reaction is going to take place. See, this is the one example. Now if you see the chiral center, from this can you mark the small, medium, large easily? You have one hydrogen, one methyl group and one phenyl. That it immediately you can understand that the incoming group has the more tendency to move towards the side of hydrogen. Okay. Now if you try to create a bulkness in this, like methyl X, the size of X should be enlarged in a such a way so that it will prefer only from the side of small group, because the difference in these three are not too much. One is hydrogen, one is methyl, one is phenyl. So the possibility is that other enantiomer may form. preferably from the side of hydrogen, but percentage of another enantiomer may be there. So you try to create now can you see the example? If your X is magnesium iodide, what is the percentage? 33. Now if you try to create X is titanium isopropoxide, the big group, okay. So if your methyl is moving, उसके पीछे जो big group है, now imagine just like a balloon, okay. आप मूव कर रहे हैं, पीछे पैराशूट बहुत काफी बड़ा है, तो उसको एंट्री करनी है, कहां से एंटर करेगा? फ्रॉम द साइड ऑफ हाइड्रोजन, स्मॉल ग्रुप. कैन यू सी द diastereomeric access change to 76%. now remove the isopropyl group with phenyl, then diastereoselectivity will be increased further. So it is your choice that how much enantioselectivity, diastereoselectivity is required to create your molecule. Okay. So in nutshell, in one word, what is the keyword? That you want to block one phase so that the other can be happen. This is the one example. Then you see this is a cyclic model. अब आपने बड़े ध्यान से देखना है साइक्लिक मॉडल क्या कह रहा है. नाउ यहाँ पे स्मॉल, लार्ज और मीडियम ग्रुप की जगह हमने क्या पुट किया हुआ है? वाई. वाई there may be some group present which have unshared pair of electrons. अब ये Y आपका क्या हो सकता है? OH group. Y can be OR, Y can be NR2. So oxygen and nitrogen पे you have a electron density. Okay. Now that electron density and the oxygen negative of carbonyl, they are going to make a chelation with the metal ion. What is metal ion? You are using Grignard reagent R Mg X. तो R ने तो कहाँ पहुंचना है? कार्बोनाइल कार्बन के पास. Mgx ने कहाँ जाना है? कार्बोनाइल के ऑक्सीजन के पास. तो एक तो आपके कार्बोनाइल की ऑक्सीजन है और एक आपके एडजेसेंट ग्रुप में भी OH या Nr2 ग्रुप प्रेजेंट है, उनके पास भी इलेक्ट्रॉन डेंसिटी होगी. ये दोनों अपनी इलेक्ट्रॉन डेंसिटी से मेटल के साथ क्या हो जाएंगे? चिलेशन. आप तो चिलेशन एंड लिगेंड्स यू नो वेरी वेल. तो जब चिलेशन हो जाएगी, दे मे ब्लॉक वन साइड. ओके, देन योर अदर साइड इज ओपन. Okay. So it depend. Now in this both example, any reaction occurring at carbonyl carbon is wholly solely depend on what? What are the environment in the adjacent? Neighbor में क्या environment है वो अब guide करेगी. और यही चीज हमने की थी फर्स्ट जनरेशन में भी और सेकंड जनरेशन में भी. Okay. So if it is a simple, there is no chelation, we call it open chain model, but if you get a chelation, we call it as a cyclic model. Okay. So these two models are more prominently going to act. Okay. Now can you see 3.11? the nucleophile is from the side of small group. Okay. So the question is, सर स्टीरियोकेमिस्ट्री कैसे ड्रॉ करेंगे फाइनल प्रोडक्ट? इफ यू कैन एक्यूरेटली डेजिग्नेट स्मॉल, मीडियम एंड लार्ज ग्रुप, you can predict the stereochemistry that which will be your most probable, most promising product in this reaction. Okay, what you have to visualize, which one is the smallest group, which have a very low electron density as compared to the other groups. Okay. So now I want to create a pure enantiospecific reaction. Now what is your plan? How I have to choose my substrate? Can you see your substrate? It has, what is the small group? You very intelligently and deliberately place small group as a hydrogen, but what is the medium and large group? Medium group is what? CH2 phenyl oxygen, and large group is what? C7H15. Now if you choose such type of molecule where the medium group and large group have bulkiness, electron density, so the 100% movement of your incoming group is from the side of smaller. So you choose smaller as a hydrogen. Now can you see the enantioselectivity? 100%, because group doesn't want to go from the side where there is C7H15 और O benzyl group. ऑक्सीजन की इलेक्ट्रॉन डेंसिटी भी है, फिनाइल की इलेक्ट्रॉन डेंसिटी भी है, so the group want to come from the side of hydrogen. So can you see उधर चिलेशन हो गई है, न्यूक्लियोफाइल देखो बिल्कुल ऑपोजिट से जा रहा है, दूसरी साइड आपने चिलेशन कर दी, बड़े ग्रुप कर दिए, फेस को बिल्कुल ही बंद कर दिया. नाउ द इनकमिंग ग्रुप इज मूविंग फ्रॉम द साइड ऑफ, there is no possibility of formation of your other enantiomer. So the reaction is enantiospecific. Okay, so it is your desirability. Now the question comes, if the same question, if you enter to the industry, they ask you as a chemist, plan a reaction. Now how you have to plan? They give you a carbonyl group, simple RCOR. Now they say this is aldehyde and ketone, try to convert into this molecule. Then what is your aim? You try to place some auxiliary in that substrate which is blocking the one phase very. अब आपने auxiliary क्या place की? हाइड्रोजन, मीथाइल, इथाइल. कितनी स्टेरिक क्राउडिंग हो पाएगी? आपने रिएक्शन परफॉर्म किया, बट आपके एनेंटियोमर क्या आए? 80-20, 70-30. देन यू यार मीथाइल एंड इथाइल करने से इतना ज़्यादा फर्क नहीं पड़ा, मुझे 100% एक एनेंटियोमर नहीं मिला. देन व्हाट यू डिड? यू ट्राई टू प्लेस O benzyl and C7H15. Now you completely masked one side, perform the reaction, and you report to your company that okay, take this pure enantioselective product. Okay, so this is your open and cyclic model which you have to see when a mechanism has to be done. Okay, so now I shown you only one example of asymmetric aldol reaction. Okay, that we have to discuss in today lecture, that how auxiliary is going to help in this case. अच्छा, do you know what is enolate? Enol form पता है क्या होता है? enol, what is enol? Tautomerism form of aldehyde or ketone. Okay, that you can convert into the enol. के लिए क्या चाहिए? अल्फा हाइड्रोजन, इनोल के लिए आपको कार्बोनाइल के एडजेसेंट में क्या चाहिए? अल्फा हाइड्रोजन. ओके, सो व्हाट यू आर टॉकिंग अबाउट, if I write this way, this bond will move here, this will come here, and this H is going to here. Okay, you are getting in carbon carbon double bond and OL group. That is your enol form. Okay, if I write in this way, you will get, this is your enol form. Okay, from this enol, if I withdraw or abstract the hydrogen from this molecule, if I abstract the hydrogen, then this is the negative charge which can be delocalized here. तो ये क्या बन गया? एक एस्टर की फॉर्म है जो इनोल से बनी है, दैट्स व्हाई इट इज कॉल्ड इनोलेट. ओके, बट इफ from aldehyde and ketone carbon carbon double bond. Can you place stereochemistry here? कार्बन कार्बन डबल बॉन्ड के अक्रॉस कोई फॉर्म पड़ी है? सिस एंड ट्रांस, जेड एंड ई. If I am talking about simple aldehyde or ketone, it is a simple. But if I am converting it into enol, that means you have a carbon carbon double bond. But if you have a carbon carbon double bond, then always you can tell that whether it is a cis or trans, whether it is a Z or E. Okay. So, keeping that in mind, if we are going to see a one reaction which I have shown you, the aldol condensation. What is aldol condensation? Which type of molecules give you aldol condensation? अगर मैं दो बेंजेल्डिहाइड मॉलिक्यूल्स को लेकर रिएक्शन प्लान करूं तो एल्डोल कंडेंसेशन हो सकती है. I required one molecule which have a alpha hydrogen, so that can convert it into a carbanion molecule. Okay. So the question is, I shown you 1, 2, 3, 4 enantiomers. How you differentiate them? 2 are syn and 2 are anti. If I have a picture in that, I can show you. Now, can you see 4.1? 4.1 में आपको क्या स्पेसिफिक नजर आ रहा है? वो ब्लैक वाले मार्कर कहाँ चले गए? करो. In 4.1, what is specific? I can check. In 4.1, you have an auxiliary which is adjacent to carbonyl group, and that auxiliary is fixed. Can you see? Auxiliary directs exclusively to the upper face of the enolate. Auxiliary ऐसे प्लेस की गई है जो अपर फेस में ही इलेक्ट्रोफाइल को आने देगी, नीचे वाले फेस में नहीं आने देगी. Now if you see your substrate adjacent to carbonyl carbon, you have a 2 hydrogen. Okay. So out of these 2 hydrogen, if I दूसरा अंदर you have placed auxiliary here which is controlling the incoming group. In your reaction scheme, what is the incoming group? Electrophile. So it is directing the electrophile to only from the upper face. Okay. Now adjacent to this carbonyl you have 2 hydrogen. Okay. Now you are adding a base. Base will abstract a proton. You will get a negative charge here. This negative charge can move here. You can get the delocalization of that. Now it is called the enolate. Now this is going to attack at the other aldehyde molecule. अब aldol condensation reaction में भी क्या होता है? If I have a 2 molecule of acetaldehyde, अगर आपके पास 2 molecules हैं, can you explain me why I have written the same molecule Can you just tell me why I have written the same molecule in two different form? That is acetaldehyde, second molecule is also acetaldehyde. In one case you are using the carbonyl of it, okay. In this case you are using the carbonyl of it. In this case, now I am adding a base. Base क्या करेगा, यहाँ से हाइड्रोजन ले गया, ये आपके पास क्या बन गया? So aldol, condensation or nomengal, benzoyne, perkene, you always need one molecule which have a alpha hydrogen. So आपने base add किया, base ने यहाँ से proton को abstract किया, you get this carbene. So this is your carbene which is going to attack. Where it is going to attack? So in this case, every reaction you have to visualize in these two form. That one side you have a carbonyl substrate, other side you have carbene. Okay. Now from this reaction, what you are making a carbene, which is going to react. Now you have chosen enol. Now see 4.2 and 4.4. If you are able to get the carbon-carbon double bond, you are always going to get fixed the stereochemistry, whether it is a Z or whether it is a E. Now can you see 4.2? The R and OM on the same side. Now can you see 4.4? R and OM on the opposite side. So that leads that what type of enolate is going to act, that whether it is a Z enolate or whether it is a E enolate. और Z and E से क्या फर्क पड़ेगा? अगर मैं आपको सिन और एंटी के केस में बात करूं, if I can show you the reaction, okay, this is a reaction. Now can you see this is asymmetric aldol reaction? You have chosen a substrate. What is the substrate? ACOH. From this substrate you required only carbonyl group. Now you required a carbene. Carbene is coming from what? E or Z. दोनों में से कोई रिएक्ट करेगा, या तो Z इनोलेट बनेगा या E इनोलेट बनेगा. तो अगर आपका Z इनोलेट बनेगा तो 50% ऑफ एंटी इनैंशोमर की पॉसिबिलिटी आपने रूल आउट कर दी, और अगर आपका E इनोलेट बनेगा तो 50% ये जो सिन इनैंशोमर्स है इसको आपने रूल आउट कर दिया. सो सच अ इंपॉर्टेंट पार्ट दैट हाउ सिलेक्टिविटी आपकी जहां पॉसिबिलिटी 25 है, आपने जस्ट एप्रोप्रिएट इनोलेट की ज्योमेट्री फिक्स की, 50% बाहर कर दिया. ओके, सो how this has to be done? First we have to see the auxiliary which has been placed AUX. That means how the auxiliary has to be generated. अब मैं just simple question है जो मुझे आंसर आपसे चाहिए, auxiliary क्यों लगाई जा रही है? हाँ जी बोलो. Blocking the one phase. ओके, सो ऑक्सिलरी में फिर क्या होना चाहिए? कल आपके उन स्टूडेंट्स ने जिनको मैंने यहाँ फ्रंट सीट पे आमंत्रित किया था, उन्होंने बहुत अच्छा क्वेश्चन एक पूछा था कि यू आर सेइंग दैट दिस इज अ कार्बोकेटाइन एंड द न्यूक्लियोफाइल इज कमिंग, you are saying you place some negative sheet here, whether this negative sheet is going to attack this carbon positive, then what is the role of that nucleophile which is incoming? So that is a very simple and very brilliant question, that what we are bringing is a reactive intermediate. but which we are hanging in between is a neutral molecule, stable entity which is just blocking the phase but doesn't participate in it. Okay. So what is auxiliary? If you go to the generation number second, आप क्या कर रहे थे? सबस्ट्रेट में ऑक्सिलरी ए स्टार लगाया, रिएक्शन किया, प्रोडक्ट बन गया, ऑक्सिलरी बाहर कर दी. तो फिर ऑक्सिलरी कैसे चूज करनी है? हमने ऑक्सिलरी ऐसे चूज करनी है कि उसमें वो कॉम्प्लीमेंट्री ग्रुप हो जो उसको सबस्ट्रेट के ऊपर प्लेस कर दे, और उसके पास वो एडिशनल ग्रुप हो जो एक फेस पे स्टेरिक क्राउडिंग कर दे पर खुद ही रिएक्शन में मत जाए. नाउ टू अंडरस्टैंड what type of auxiliary you should choose. Number 1, the auxiliary should be able to attach to the substrate. Second, in any of the phase, auxiliary just block the phase by creating the steric bulk, electrostatic interactions, so that the incoming reactive intermediate should not enter. But that doesn't mean the auxiliary itself start participating in the reaction. Auxiliary has to do what? Either blocking or making a chelation, so that one phase is blocked. Okay. Now in this case, if you see, this is SAMP and RAMP. हाइड्राजीन मॉलिक्यूल क्या होता है? NH2 NH2, वेरी सिंपल. सो यहां पे आपको एक नीचे NH2 दिख रहा है, उसके बाद जो नेक्स्ट नाइट्रोजन है उसमें भी 2 हाइड्रोजन चाहिए. उन 2 हाइड्रोजन की जगह पे यू प्लेस एनी साइक्लिक मॉलिक्यूल. ठीक है. व्हाट इज हाइड्राजीन? NH2 one NH2 group is required to attach with the substrate, so it is a complementary group. The other NH2 group, you replace both the hydrogen with some cyclic structure, and that cyclic structure must have some group which have a electron density. You can you see, you have a O methoxy, the oxygen has a unshared pair of electron. So such type of enantiomers of hydrazine कि हाइड्राजीन का फर्दर डेरिवेटिव बना दिया ताकि हाइड्राजीन के एक NH2 का यूज कर लो ऑक्सिलरी को सबस्ट्रेट के साथ हुक करने के लिए. नेक्स्ट सब्स्टिट्यूटेड ग्रुप लगा दो जो स्पेस में ऐसे हैंगिंग करे, एक फेस में इलेक्ट्रॉन डेंसिटी प्रोवाइड करे. सो यू हैव अ बोथ द enantiomer, the S for one enantiomer, R for the another enantiomer. So ये एक ऑक्सिलरी है, इसकी मैं सिंथेसिस पे नहीं जाऊंगा. ओके, these are the different steps that अब आप देख रहे हो स्टार्टिंग में 1 and 2 में जो आपने कायरल यूनिट देख रहे हो वो स्टार्टिंग कायरल पूल से ली गई हैं. आप इन पूरे रिएक्शंस को अगर ध्यान से देखो, कायरल यूनिट कहीं पे भी सिंथेसाइज नहीं की गई. It has been taken from the pool. If you see in case of first, that is taken S proline. S proline में लेके आपने सैंप बना दिया, फिर R ग्लूटामिक एसिड जहां कॉन्फ़िगरेशन R है उसको लेकर आपने आगे रैंप बना दिया. तो you don't have to go to synthesis of it. Now try to figure out the reaction. इस मैकेनिज्म को आपने ध्यान से देखना है. समटाइम्स द फेस इज ब्लॉक्ड, समटाइम्स द ऑक्सिलरी इज आल्सो हेल्पिंग टू ब्रिंग द इनकमिंग ग्रुप. मैंने कल आपको एक एग्जांपल दिया था ना, इफ यू नो समबडी यू कैन ब्रिंग दैट पर्सन टू मी, बट इफ दैट पर्सन इज रोमिंग अराउंड आई एम नॉट गोइंग टू इंटरेक्ट विद देम. ओके, so I can say someone is coming to me but you people are in between. You can say it is a blockage, but maybe you are trying to interact that person more comfortably with me. So it is not always if auxiliary is placed it is blocking, sometimes it is also helping to bringing the incoming group close to it because of the other functional groups present in it. Now if I say what is the first reaction, what is your substrate? RCOCH2 R prime. So आप करने जा रहे हो रिएक्शन अल्फा एल्काइलेशन ऑफ कार्बोनाइल ग्रुप. व्हाट इज अल्फा एल्काइलेशन? कार्बोनाइल के अल्फा पोजीशन के एक हाइड्रोजन को आप रिमूव करके नया ग्रुप इंट्रोड्यूस कर रहे हैं. दैट इज योर अल्फा एल्काइलेशन. अब आप बताओ अगर मैं वन हाइड्रोजन को रिमूव कर दूं तो वहां पे कायरलिटी जनरेट हो जाएगी. RCOCH2 R प्राइम. R प्राइम अलग ग्रुप है, हाइड्रोजन अलग ग्रुप है, RCO डिफरेंट ग्रुप है. नाउ वन ऑफ द हाइड्रोजन इज ब्रिंग बाय R डबल प्राइम, यू गेट अ कायरल सेंटर. सो दिस इज द ऑब्जेक्टिव विच वी हैव टू डू. ओके, सो उस ऑब्जेक्टिव के लिए फर्स्ट व्हाट वी हैव टू डू, वी हैव टू रिएक्ट द सबस्ट्रेट विद ऑक्सिलरी. तो ऑक्सिलरी में कॉम्प्लीमेंट्री ग्रुप क्या है आपके पास? NH2. तो कार्बोनाइल ग्रुप को NH2 से आप रिएक्ट करोगे तो क्या बनेगा आपका? कार्बन नाइट्रोजन शिफ्ट्स. नाउ इफ यू सी द फर्स्ट स्टेप, you are treating your substrate with the auxiliary which is the same, you are going to get only carbon nitrogen double bond. That is a simple reaction of aldehyde with the amine to get Schiff's base. Okay, now if you see in next step, you add a base, you add LDA as a base. So that base abstracted a proton creating a carbon negative, that is the carbanion molecule. Now place the important part of the auxiliary. Now can you see that this negative charge and negative charge of the unshared pair of the oxygen, they act from the both sides to bind with the lithium positive. That's mean they are doing a chelation. Now you have add the group. Now the third part, you are bringing the reaction. मैं आपको इसको एक direct reaction में दिखा रहा हूँ. See the picture. Now can you see in first step is a very simple reaction of organic chemistry like +2 level. You are adding aldehyde and amine, get carbon nitrogen डबल बॉन्ड. इफ यू सी इन योर फर्स्ट स्टेप, आपका NH2 और कार्बोनाइल ऑक्सीजन रिएक्ट कर रहे हैं, कार्बन नाइट्रोजन डबल बॉन्ड बन रहा है. फिर आपने बेस ऐड कर दिया. एडिंग अ बेस, you abstract the proton from the alpha position and you create a negative charge. Now if you see the three dimensional picture, chelation हो गई. अब आप अपना इनकमिंग ग्रुप लेके आ रहे हो. व्हाट इज इनकमिंग ग्रुप? R डबल प्राइम X. अब जनरली तो बात ये है, नीचे तो सारी क्या हो गई है? चिलेशन. अपर फेस इज फुल्ली ओपन, so R double prime should come from the upper face. As we are talking, the auxiliary is blocking the face, the R prime should come from the upper side. But can you see in this, R prime X is coming from this side? Why this change occur? Because when R prime X is moving either on the space above or space below to enter into the reaction, it has R double prime X. This X is coordinated with lithium. अब आप इमेजिन करो इस रिएक्शन में आपने ये ट्रांजिशन, बड़ा सिंपल है, अगर आप अटेंटिवली देखोगे ये आपकी ट्रांजिशन स्टेट है. ओके, यहाँ पे आपके पास अब बाहर से आ रहा है कुछ, दैट इज R डबल प्राइम X. वो ऐसे ऊपर से भी आ रहा है, नीचे से भी आ रहा है, फ्रीली मूव कर रहा है. उसे तो कार्बन के पास लैंड करना है. तो क्या है, ऐसे वो ऊपर आ रहा है स्पेस में, नीचे आ रहा था, उस X को क्या मिल गया, क्या दिख गया नीचे? लिथियम पॉजिटिव. अगर वो लिथियम पॉजिटिव है, X नेगेटिव है, व्हाट दे ट्राई टू डू, दे ट्राई टू इंटरैक्ट. सो क्या हुआ, ये जो R डबल प्राइम X है, X जैसे ही इसके साथ इंटरैक्ट हुआ लोअर फेस में, तो अब ये मूव तो नहीं कर सकता, तो R प्राइम लोअर फेस से ही इस कार्बन की तरफ अप्रोच कर गया. नाउ व्हाट आई एम सेइंग दैट यहाँ पे इफ दिस इज मी, दिस R प्राइम इज कमिंग आउटसाइड to get signature in a form, it is moving up and down, I am not going to interact. That is the you have here. You interact with me, sir, this is my friend, this is my brother, this is my sister, please get attested his or her certificate. So okay, I know you, okay, bring it, I sign it. But if he or she is roaming, it is my purview that I want to interact or no. So sometime chelation also help to make the group. Now if this lithium positive is not present here, only O methoxy is there, then O के इलेक्ट्रॉन डेंसिटी और X की इलेक्ट्रॉन डेंसिटी आपस में आने देते एक दूसरे को, will it allow? जो OM ऑक्सीजन के अनशेयर्ड पेयर्ड ऑफ इलेक्ट्रॉन्स है और X के अनशेयर्ड पेयर्ड ऑफ इलेक्ट्रॉन्स है, क्या ये एक दूसरे के पास एक फेस में आ सकते हैं? इनकी जो इलेक्ट्रॉन डेंसिटी थी वो दोनों ही किसने ले ली, लिथियम पॉजिटिव ने, टू मेक अ बॉन्ड. सो योर R प्राइम इज फ्रीली अवेलेबल फ्रॉम दिस साइड टू एंटर इनटू द रिएक्शन. ओके, सो this is the important part that which we have discussed, how chiral auxiliary is helpful to prepare a one type of enantiomer. अब आप अगर आपके माइंड में इस ऑक्सिलरी का रोल नहीं है और आपके माइंड में सिर्फ एक ये 3 dimension पिक्चर आपको नजर आ रही है प्लेन में, what you are going to accept, R डबल प्राइम X आ रहा है, ऊपर का फेस तो इस कार्बन पे अटैक करना है, ये ऊपर का फेस पूरा खाली पड़ा हुआ है, फॉरगेट अबाउट दिस ऑक्सिलरी, ऊपर का फेस खाली है, नीचे का फेस खाली है, 50-50% इज द प्रोडक्ट, दैट इज योर racemic mixture. But how this racemic mixture is turned down into a enantiospecific reaction by just placing a auxiliary, which itself is not going to enter. Once the reaction is over, you are going to cut this carbon nitrogen bond. कैसे करेंगे हम? शिप बेस की हाइड्रोलिसिस कर देंगे, वापस आपके पास क्या आ जाएगा, अमीन एंड एल्डिहाइड. But very selectively you are able to place this R double prime from below the plane. So how this auxiliary actually helping to place the R double prime from below the plane if your drug molecule required this. So आप सिंथेसिस का प्लान करने जा रहे हैं तो सिंपल रिएक्शन है, आपके पास एक कार्बोनाइल कंपाउंड है जिसका आपको वो देंगे, दिस इज द सबस्ट्रेट, दिस इज द रिएजेंट, प्लीज मेक दिस प्रोडक्ट. सो यू हैव टू सी, ओके, दिस इज इन अ प्लेन, इफ आई एम एडिंग R डबल प्राइम X, इट कैन गो फ्रॉम अप साइड, इट कैन गो फ्रॉम लोअर साइड, आई एम गोइंग टू गेट अ रेसमिक मिक्सचर, देन मुझे रेजोल्यूशन करनी पड़ेगी, 0.001% भी एक मुझे HPLC करनी पड़ेगी. अगर 0.011% भी एक एनेंशियोमर दूसरे में आ गया, इट विल नॉट बी एक्सेप्टेड. सो यू डिड वेरी स्मार्टली वन थिंग, ओके, फर्स्ट ट्राई टू हुक वन ऑक्सिलरी. Once you hook the auxiliary, it is a simple shift based reaction. You add the auxiliary, now auxiliary will take care of the entry of the incoming group. Once you place a group from only one phase, remove the auxiliary. Now you don't have to go for any separation, you don't have to go for any column chromatography, you don't have to go for any HPLC purity, because you get enantio specific product. So how smartly you choose the auxiliary, okay. So there are a number of auxiliaries are available, SAMP, RAM, Sultam, Evans, Mazum. हर साइंटिस्ट जो आया उसने एक नया ऑक्सिलरी को डेवलप किया एंड दे ट्राई टू एंटर इनटू द so is it now okay that how auxiliary work, how to synthesize the auxiliary, that what is the smartness of any auxiliary? Now can you explain me in 2 points जिसमें नेक्स्ट रिएक्शंस है वो हम अपने आफ्टर लंच वाले में करेंगे. बट नाउ अ वेरी सिंपल क्वेश्चन कम्स इन अ यूजीसी नेट एग्जामिनेशन, व्हाट इज सो स्पेसिफिक इन ऑक्सिलरी, क्या होना चाहिए ऑक्सिलरी में, और इफ यू आर प्लानिंग टू सिंथेसाइज एनी ऑक्सिलरी व्हाट फीचर यू फाइंड इन ऑक्सिलरी. For asymmetric question will be in a way, for any asymmetric reaction how you choose a auxiliary. So you write number 1, it must have some complementary functional group to interact with the substrate and it should come back after the reaction. ऐसा नहीं है आपने कॉम्प्लीमेंट्री ग्रुप चूज किया, उसने सबस्ट्रेट के साथ बॉन्ड तो बना लिया, बाद में आप उसको ब्रेक ही ना कर पाओ. It should be a vice versa position. Second, it must have some functionality which may help to create a chelation, to block the one phase, or a group which will allow to enter the incoming group from only one phase. These two special features are required for auxiliary, otherwise auxiliary has no use. Clear हो गया? So अगर वो कहते हैं आपने ऑक्सिलरी ना रू ली होती तो ये रिएक्शन सीधा ही कर लिया होता. आपके पास RCOCH2R प्राइम है, यू परफॉर्म अ रिएक्शन, ओके वी परफॉर्म अ रिएक्शन बट वी गेट 50-50. If we don't have any auxiliary, you have a planar carbonyl carbon, the R double prime X is coming, it has nothing to face from both the side. तो 50% वो ऊपर से आ जाएगा, कोई रेट ऑफ रिएक्शन उतना ही फास्ट चलेगा, दोनों ट्रांजिशन स्टेट में कोई डिफरेंस नहीं है. 50% फ्रॉम अबव साइड, 50% फ्रॉम बिलो, यू गेट अ racemic mixture. So if you required both enantiomers, then you need not to go for auxiliary. But if you required selectively, अब आपको इस केस में आ गया एग्जाम्पल कि R प्राइम नीचे से आ गया फ्रॉम द लोअर फेज, नाउ आई वांट टू गेट फ्रॉम द अपर फेज, तो यहां आपने सैम्प चूज किया, उसमें क्या चूज करोगे, रैम्प. फॉर बोथ साइड यू हैव अ बोथ ऑक्सिलरीज. क्लियर? ओके. You miss the important point in this which we are talking, it is coming from the lower side where there is a crowding, the R prime axis coming from the same side, because in this case the X negative or R incoming group is interacting with this lithium. So maybe there is a crowdness, maybe there is a blocking of phase, but sometimes that blocking may helpful to deliver the group from the same side because it is allowing to enter from its. Okay, so मैं आपको बाद में बता दूंगा. Okay, ठीक है ना. मैं ये पेन ड्राइव निकाल दें.
Thank you so much, sir. This was indeed a very informative session. We will resume the next session in 5 minutes. We now move on to our next lecture by our distinguished expert Professor Sanjay V. Malhotra from Oregon Health and Science University. He will be presenting on molecular imaging in drug development with a focus on introduction to multimodality molecular imaging. This session will take us into the exciting world where chemistry meets imagination and innovation.
Good morning. All right, so far, so we talked about, experimental therapeutics, what all it takes to be part of experimental therapeutics. It's a multidisciplinary process, as you just saw. There are many, many disciplines involved. Now, when we do an assay of just petri dish. So basically, you take a drug, a small molecule, whatever you do, you're just studying biology. You have to know that it is correct or not. So previously, going back to, there were simple chemistry methods that came to understand the method that was in 18th and 19th century. Then came instrumentation method like chromatography, NMR, IR, all these to understand the molecules. NMR, I might have mentioned, NMR led to MRI, magnetic resonance imaging, जो hospitals में करते हैं, that came out of NMR. NMR came out first, and that technology then led to MRI. We use various techniques, we use various methods, and in order to make sure that we can follow what we are interested in. One of the techniques, for example, when the optics physics became more and more advanced, imaging, that when you light various rays of wavelength, when you light on something, it reflects depending on where the angle is, all of that you can measure and can get sense of the target. Imaging similarly is a modality which is very, very important now for the last few decades, and especially more and more advanced now, where imaging is used to be able to see कि क्या हो रहा है, whether it's inside your petri dish when you do, because for example, as I was telling you, I showed you on this side that you can make different cell lines. You can create cells, you can create vectors, because you have base pairs based on paper. For example, let's say if you have any cancer type that you want to understand and develop the new drug, new therapies for that. I'm focusing on therapy side because experimental therapeutic is more focused. The way we have designed this course is more focused on the therapeutic side. But you want to learn more about the biology. You want to understand what is going on inside whatever your system is, whether it's a cell, in vivo, or humans. Uske andar ho kya raha hai. So when you create vector, so I'm starting from a very simplified version. So if you have to create a cell line, you create cell line, and I was telling you what you do is at the end you can put a fluorescent, luminescent, some tag to that, and then that tag will light up, and based on the intensity of the tag you can see कि उसके ऊपर कितना effect है. You can actually quantify. आपने ड्रग लिया, ड्रग से ट्रीट किया, whether it's 15%, 18%, 80%, 90%, based on the intensity. कहाँ से पता चला कि 80% है, किसने कहा कि 18% है, right? There has to be a way to quantify. Okay, that was, and you can actually image. Now you probably are, I'm sure, familiar with or have seen, when you go to physician, MRI is a technique. When you go to the physician, or physician सारा लेते हैं, X-ray क्या है? It's image, right? जो image लेते हैं, that imaging using X-ray as technique that has come out, right? So imaging has become extremely, extremely important process in order to see what is actually going on. उसके अलावा तो indirect methods होती थी पहले. Imaging helps to know if there is a drug is affecting, for example, if there is a tumor type inside, be able to image to see what is actually where the tumor is, so surgeon can take out that tumor, the surgery which is done first. if you're using radiation, if you're doing laser, be able to see wo hai kahan. But how do you see that? So through imaging. So imaging techniques, various imaging techniques have become extremely, extremely important these days, right? Same way, now this is, I'm talking about patient, now trace back, same thing inside animal. If you're developing something, you're trying to understand, you need to know what is happening. So imaging, and there are various modalities जो हैं imaging के basis पे have come out. So I will just quickly give you an introduction of. So I have highlighted these, and as I was telling you, I have taken notes in such a way so that once you review that, you will have more information. So it doesn't mean that your text has, or you have to, just go through text, but because I think the plan is that I will give all my slides to Dr. Gurjaspreet Singh, and then you will have access to that. So when you see a lot of text on my screen, personally, my presentation style is I don't like text. How आपने सुनी ना, picture worth 1,000 words. Picture conveys the message more, but sometimes it becomes necessary. So you'll see on my slides, some of my slides, उसमें text होगा. जैसे कल रात को बैठ के भी मैंने उसके ऊपर yellow किया कि you don't read the whole thing, but read the yellow part. That's the important part. So anyway, why and how these modalities are helpful. As I was telling you yesterday, I showed one of the slide I had was where you see thousands, just say pick a number like 10,000 compounds go through, and maybe one will see the light of the commercial world. It's an extremely tedious, multidisciplinary, and extremely expensive process. Any and every technique that can help us to cut down the cost, the time, the resources, the frustration is a positive step in that direction, imaging being one of those. So it's a very time sensitive. So what new strategies are needed? Imaging basically helps in characterization of molecule level, protein level, about the cell lines, whether animal, you can image the animal. If you're doing something, image. So when you do, you actually see whether cross kar raha hai ki nahi kar raha. Yesterday, for example, now blood-brain barrier, how do you measure that? Whether it crossed or not. You can image to see whether the effect of the drug happened or not. Uski jo dawa jo hai, if it's tumor, you can see the tumor shrink hua, how to measure that, how to before or after treatment. So imaging is an extremely so these are in radiology departments. I'm sure PGI may be had all radiology departments. This is a routine process. सुबह से शाम तक वही करते हैं, scans करते हैं, scan को पढ़ते हैं, scan को समझते हैं, then they do the measurement and tell के patient को क्या है, कितना असर हुआ, नहीं हुआ, कहाँ है body में, जो भी या organ में जहाँ है. So it is not just predicting the clinical observations, but based of that you can use that information even to understand the mechanism, how it might be happening. Because if you assume that there is something, it is happening certain ways. Now there are many, and I'll come to what is happening in my institute, for example. So it has, as you can just imagine, पहले कुछ नहीं था. You do the analysis. Once it's done, you use indirect methods or invasive methods. These are non-invasive. You don't have to insert something inside the animal's body or human body. These are non-invasive. They don't invade inside. So, and you can just take a picture. So therefore, these are extremely powerful, extremely helpful, and extremely important. And that's the reason they are routinely used, because patient को कुछ भी inject नहीं करना पड़ता, कुछ नहीं पड़ता, खिलाना नहीं पड़ता, पिलाना नहीं पड़ता, but you can just simply take the image उसके साथ में. And in most non-NCDs, non-communicable diseases में बहुत application है, whether you say cancer, cardiovascular, neuro, Alzheimer में. There are not many methods available, unfortunately. Neuroscience is a very fast developing field and unknown. So again, point being, if you want to pursue a career, be an expert, just pick, as a chemist, pick any one of these areas and just follow. And before you know, there's ocean and ocean of knowledge and ocean of unknown. So you can pick up an expert धीरे धीरे करके in one field, and before you know, you'll become an expert in that area. Just pick one area. Problem, I mean, there is so much unknown when it comes to the biological creation. So the I mean, many these phrases have been used in the past. You pick one protein, it will take you end of the life. एक प्रोटीन को समझने के लिए आप अपनी सारी ज़िंदगी लगा सकते हैं, फिर भी नहीं समझ पाएंगे, बट उसके अंदर आप मास्टर बन जाएंगे, उसके अंदर आप नाम कमा सकते हैं, क्योंकि आप सबसे पहले करने वाले होंगे जो उसको बताएंगे. So there is so much to be done. So imaging, for example, as a chemist, pick one. Imaging agents बनते हैं, and I'll come to that briefly. So again, you know, it's an imaging thing which is used in the drug development from early assessment of target, protein को समझने के बारे में, drug development candidates जिस तरीके से बनाने हैं, and of course phase trials में, phase 1 to phase 3, again, depending on that, radiology can take the image, and I'm again using this for either neuron cancer or cardiovascular जो NCDs हैं. This is a routine thing, and as I said, it's an expensive process, but imaging, जिस मर्जी चीज़ की, any new technique, any new modality that can help in better understanding is a positive thing. So here is summary of various modalities which are used. Optical imaging, सिर्फ laser, सिर्फ light उस पर डाल के, green light, you get the sense. So there are, each technique, for example, has strength and also have limitations. Some, but strength can be used for whatever your goal is, whatever you're understanding in. Optical devices are very helpful in for example, high throughput scanning. So in my lab, we use very regularly. You take, when you do high throughput scanning, when you screen thousands and thousands of compound against a single protein to see कि कौन सा compound काम करता है, when you treat with that, you take the cell line, cancer cell lines, you can and put it in 96 well plate, 384 well plate, or 1536. एक ही plate के अंदर 1536 wells हैं. You can put your cells, you can put 1536 drugs at a time. So we, for example, if anybody has to do like 250,000, 250,000 के करीब अगर टेस्ट करने हैं, फटाफट प्लेट्स में ट्रीट करते हैं, then imaging, optical imaging can quickly help to see कि उसका असर कौन से वेल में, कौन से कंपाउंड ने कितना किया, and then from that large collection you can pick कि उसमें से कौन से hits हैं. So imaging is used there. Magnetic resonance, easy, advantage is clinical translational, but it is a costly process. Ultrasound is used commonly. Use again, clinical. It's a low cost, but it depends on the operator. There is a depth only up to which that UV radiation can go. अगर डीपर इनसाइड द बॉडी अगर करना है तो वो उसको पेनिट्रेट करके वहाँ जाएगा नहीं, तो यू कैन नॉट यूज़ दैट. यू कैन यूज़ इट फॉर द सरफेस के आसपास तक ही, राइट. बट सरफेस के आसपास तक है, डिपेंडिंग ऑन उसकी यूवी रेडिएशन कितने तक जा सकती है, उसको आपको रिफ्लेक्शंस तो देगा. बट दैन द एडवांटेजेस कि वहाँ तक जाता है, डिसएडवांटेजेस कि उससे डीपर नहीं जा सकता, तो you use as is needed. PET imaging, very commonly used, is costly, but it is one of the most commonly used imaging technique by radiologists. SPECT, another form of so PET is positron emission topography. If you see that, and then CT is again high spatial resolution, but it's not specific to target. It can give you full spatial resolution, but not specific to a target. So each modality has its own characteristic which can be used, which can be employed for एक मिनट, I'll just change the mode. Just give me a second. It is not in the presentation mode, and I'm trying to see how I'm trying to put in that presentation mode. No, it's not in the presentation mode. Huh? यहाँ पे view, under view, it should show here. Slide master presentation view. Let's see. ये जो है, when I do this,
Here it should be the notes part.
it should be the notes section. Yes, now it's better. Thank you. Okay, so okay, so this is again, you know, in as I was telling you, many ways by you where imaging we do this routinely. For example, all those who do the lot of in vivo work, we use this, for example, routinely. When a mouse studies are, for example, done, you inject whether it's a green GPF, green protein, or to tag as the optical imaging के part के लिए, and then you can from there also your distribution of your drug inside where it went. If it didn't went, you can simply take the images of that. Now this is what I was telling you. You can do actually even mice high throughput. There are ways to do where you can actually just tell them, for this is an example of a receptor. So if you want to identify a receptor or a protein, so receptor, let's say उसके right flank thigh में you inject the cell line, and you are interested to see in that tumor part what actually is happening. You can use imaging as a technique to learn more about various different, you know, ये जैसे receptor ligand interactions, whether it happened or it didn't happen. So this is tumor again. In the tumor size, to see what is the tumor size, you measure the tumor size. Now there are multiple advantages, as I mentioned, for surgeon to be able to see where to see the surgery, how much portion, what to do surgery. For a researcher to be able to see before and after what happened. If you're treating, is the tumor shrinking? What is the size of the tumor? What is the volume? You can measure you can measure all of that, and if it is shrinking or not, it tells you. So imaging can be used rather than having to just poke it out and take it out. Instead of having to euthanize the animal, you can actually, through imaging, keep on constantly giving your treatment and see how much and what the effect is happening. So again, imaging, again a powerful. Now clinically, as I was telling you, for clinically, there are many applications for identifying if there is a target or not, for distribution, if the drug is given or if something is happening, how it is distributed in various organs, whether it goes to the liver, intestine, stomach, heart, lung. You can take the imaging and you can just see whether the drug was distributed or not, instead of having to poke and take it out any pieces. Simply just the image, and you can follow that. You can put a tag and you can follow the tag and see by simple imaging it can help you. The pharmacodynamics, what happened, what drug does to the body. Remember, pharmacokinetics is what body does to the drug. Pharmacodynamic is what drug does to the body. Again, shrink hua, nahi hua, kitna hua. And again, patient selection, patient stratification. Even there, imaging is used to see whether the patient is the right patient or not for the treatment. Now there are, how do you do that? So basically for positron emission topography, there are, now how do you do that? As a chemist, think for a second. Aapka neutral compound hai, does not have anything, so how do you do that? What will give the signal? So there are radioisotopes. Oxygen hai, to instead of giving oxygen 16, you can give, as you see here, oxygen 15, carbon 11, your gallium 68, fluorine 18 is one of the most commonly used compound in the world is F18. So in our university we have our own cyclotron, so I oversee that center also, Radio for Pharmaceutical Research. So what we do there is we design tags, we design for many our researchers, whether they're in any field, cancer, neuroscientists or cardiovascular imaging, jo bhi jahan bhi chahiye, and even monkey colonies, they all come to our lab, they all come to our center, whatever they need. So if they design a ligand which they think is actually affecting, now what you need to do is you need to add one of these tag, fluorine tag, fluorine 18 हुआ, gallium, oxygen, carbon, you attach to their molecule of interest. They inject, they're simply through imaging, then they follow. Okay, treatment is happening or not. So this is a huge field now. It's a very big field. So again, it's purely chemistry. So in my radiopharmaceutical center, I have a radiosynthetic chemist, and it's a highly demanding, they are very much in demand because there are not that many. So if you want to become a radio chemist, demands are high. And I pay them 3 times more than I pay to my chemists, because they have to be licensed. They take their special license, but then they are highly in demand because nobody knows how to work in the radionuclide. These are radionuclide. But anyway, these are the applications. So you tag whatever the molecule of interest a scientist has for whatever reason, for understanding that, for the treatment part, for simply understanding the mechanism, understanding the biology. But so the scientist comes to us as radiochemists and says, this is the molecule I'm working on, this is the antibody I'm working on, this is the RNA I'm working on, or antisense I'm working on. It works in something. Now I'm interested to see how much it in fact has inside the animal. But I want to do very quantification. So our radiochemist will take that, whatever the molecule of interest, they do the whole synthesis process, but in somewhere in the synthesis process, they will insert one of these guys. That's one part to understand. How to select, which one to select, is dependent on also on the half-life of the radionuclide. So if the half-life is only oxygen 2.1 minute, just imagine आपने बनाया और जब तक hospital पहुंचोगे उसकी half-life तो खत्म हो गई, right? So what you do is you make a large quantity of that. So by the time it gets to the, अगर आपने बनाया है 10 मिलीग्राम, हर 2.1 के अंदर वो 5 हो जाएगा, 2.5 हो जाएगा, सवा हो जाएगा, वो होता जाएगा. So you make a large quantity, and if that's the only thing which is visible. And therefore, for example, in our many places, it's like I said, there are many these radio pharmacies, as they are called, radio pharmacies people. And then the carbon 11, 20 minutes, 20.4 is the half-life, right? Gallium 68, so that gives you still. So हमारा जो monkey colony है, it is about 25 minutes around away from our radiopharmaceutical center. So you make enough of that. We have a courier service ready. That's all they do. The courier service का full-time job है. When it's needed, they simply जाता है और एकदम lead उसके अंदर तक, because you cannot take the radionuclide outside. They have to be protected to the body. Put it inside, उसके बड़े jar होते हैं, lead-based jar. Put it in the jar. Courier rushes to the labs which are in the monkey colony. They do that, but we make enough of that. They can do the treatment. 68 minutes उसका half-life period है. And same way, fluorine. This is why this is, as I mentioned, FDG जो है is the common. If you Google, if you are sitting to FDG, F18, this is the most commonly used. क्यों? क्योंकि उसकी half-life लंबी है. आपने बना के दूर तक पहुंचा सकते हैं. And literally, many times, depending on, so for example, we are south of Seattle and north of San Francisco के बीच में हैं. So depending on, so British Columbia, state of Washington, Oregon, California, and on the east side, then you have Idaho, Montana, दूर hospitals, and they want to do the treat. There are helicopter services. If that is what is needed, helicopter बनता है, फटाफट जाता है, helicopter will fly it there, take it to them, wherever the treatment has to happen. But all things, that's the service part. But to design, these are taken into account. Okay, it has enough of half-life to get to. So for patients, for example, they are, so we are, so before, I'm at Oregon Health and Science University now, but just before that I was at Stanford. And even though I do some work there, but these big universities, they have their own cyclotrons. Otherwise, most university they have to just get from जैसे आप vendors outreach वगैरह से लेते हो मंगवाना पड़ता है उनसे. So we have that advantage, and that gives the leg up in the sense that our investigators can think of doing research in this area a lot. But again, these are things. So you can put any of these, and there are more. There is a whole list. I'm just showing you four, just to give an idea okay, how and what factors are logistically, practically important. उसकी half life क्या है देने के लिए. Same way there is nitrogen. Cardiovascular studies के लिए nitrogen बहुत is commonly used. Cardiovascular imaging के लिए. So our center, for example, is, if this is the center, associated building, attached building next to is the cardiovascular, and maybe जहाँ से enter करते हैं, not too far from here, maybe 100 yard, no, not 100 yard is too big, 50 yard is our hospital, 4-tier hospital. So idea is location is such so that you quickly it goes to for patient के लिए जाना, जिन्होंने patient treatment के लिए, physicians they need, doctors they need for the patient. So that goes there. Now again for that, there are regulatory environment कि कितना clean होना चाहिए. आप when we go inside the radio pharmacy, आप ऐसे नहीं यहाँ चलेंगे. You have to be like spaceship. इतना tight नहीं होता, but you have to be fully, because and you have to be absolutely हाथ साफ किए हो, अपने gloves पहन के you go there, because highly sensitive part, right? But this is how the field has changed tremendously. Now, if you guys are interested to get into therapy, radiotherapy is presumably going to be a new frontier in therapeutic drug development. And if you want to build your career, if you are interested in, think of radiopharmaceuticals. There is a lot of investment happening. You don't need to worry about that. There is billions and billions of dollars are being invested क्योंकि उस field को आगे लेके जाने के लिए. The idea being कि जो chemotherapy है उसके side effect हैं. Other modes of therapies, there are side effects. What if you can come up with a radiopharmaceutical, radiotherapeutics, for example? So radiology भी करता है, and also it's a therapeutics. It can have a paradigm shift, but that's the field is evolving. So if you are at an early stage, think of doing your PhD in one of those areas. Those who are have done PhD in chemistry, think of doing your postdoc in those areas. If you show interest in, anyway, I think that certainly came to my mind. If you are really interested in doing that, apply to radiopharmaceutical labs. As a chemist, you apply that you are interested in that, because not many people will go into that route because everybody wants to go to pharma industry. But radiopharmaceutical के अंदर आप करते हैं, it's a very niche area, it's a very limited area, but the market is booming. And like I said, I paid 4 times more than I pay to my chemist. So scientifically, it's an extremely important area and rapidly growing field of research. So I mentioned about MRI. So basically, weak magnetic dipole, you know how the NMR works. Atoms, electrons, how they are aligned depending on what their polarity is. It splits. You look at that, 3-dimensional, look at the J constant, look at the splitting, and you can identify. So similar principles are applied to imaging and for body imaging. And now, advantages. The body has aquatic water environment, so hydrogen oxygen bonds easier, not too difficult, and they are not interfering into the weak low energy. So you can easily determine the concentration of hydrogen atom, how much water or a fat, and their local environment. You can easily measure that. And then that's the functional application of MRI. This is what I was trying to tell you. You can study the biodistribution. if you have an agent, and now just imagine to that agent you have F18 for example, उसमें tagged है, it is being distributed. You through imaging you can see whether it went to liver, or it went to intestine, it went to any other part of the body. वो imaging से आपको बता देगा. So you don't have to worry about. So imaging, as you can see, is extremely non-invasive. आपने कोई सुई नहीं चोबनी उसमें. You can just simply take the picture and it can tell you that target interaction, whether your target for which the dose that you are giving, whether the dose is reaching to the target or not, you can, by imaging, you can see कि उसका असर हुआ कि नहीं हुआ. Right. Now, at my institute, in actually one of my colleagues, she is an expert, she's a chemist, but she started focusing on neuroimaging. So she designs, she makes chemicals which are fluorescent, specific those molecules that they have the properties that they can be fluorescent. One of the challenges in the body is to cross the blood-brain barrier. ब्रेन के अंदर कैसे करना. जब सर्जरी कॉम्प्लिकेटेड सर्जरी होती है तो सर्जन हैज that's why you, well, I don't know how it is here, and I'm assuming that too, you take second opinion, third opinion, fourth opinion before you decide on which surgeon to do that. The reason for that is the person has to be very experienced. These are very delicate nerves जब करते हैं ऑपरेशन सर्जरी करते हैं, और doctor is basically opening and the best of knowledge and skill that person has is trying to do that. Now just imagine if there was a chemical, harmless chemicals, but it is fluorescent, जिसकी imaging कर सकते हैं. वो chemical दिया गया हो, अब clear cut black and white picture, डॉक्टर की इतनी बड़ी स्क्रीन है, वॉल साइज की स्क्रीन है, और पेशेंट के सामने वो एग्जैक्टली देख सकता है, बिकॉज दैट केमिकल इज क्लियरली गाइडिंग कि नर्व के अंदर जा रहा है कि नहीं जा रहा, और कौन सी नर्व को काटना, गलती से किसी दूसरी नर्व को ना काट दे. पहले ये सब नहीं होता था. दे हैड टू टेक देयर बेस्ट ऑफ their skill set, imagination, whatever was available at the time. And now these chemicals are extremely helpful in surgery for surgeons. So you inject those images, उसकी imaging आ जाती है, you take the imaging, literally, I'm sure यहाँ पे भी होगा, in surgery room, वो छोटी छोटी monitor नहीं, they are large screen जो एकदम जहाँ पे जो surgeon काम कर रहा है उसके सामने होती है. It's now अब तो, I mean, our institute has, I don't know how it is here at PGI or AIMS or other places, robotic surgery. And why robotic surgery is helpful? Because of the imaging. It has opened up in field because imaging, you can see on the image what doctor has to do, what surgeon has to do. So just imagine imaging how much it has changed compared to just imagine कि वो है ही नहीं, where it was to where it is now. So imaging, not just for the treatment, for the therapeutic purposes, it helps to understand in many, many ways at the molecular level, at the organ level. Again, as I was saying, pharmacodynamics, what drug did to your body. If it's a tumor size, if you give tumor, what happens is inside the tumor, the proteins have specific, inside the tumor, environment is different. TME, tumor microenvironment, is different than the normal cells. In there, the glucose levels, the oxygen levels are different. You know the term hypoxia, lack of oxygen, right? In there, if you have chemicals that light up in that environment, you can easily see कि उसका size क्या है, whatever that be. Not necessarily cancer, but if there is a nerve rupture, you can see where the rupturing happened. So biodistribution, and same way pharmacodynamics, if you treat with something, you take the size before, you look at the picture before, and during the treatment process you can keep on taking, and you can tell you whether effect उसका हो रहा है या नहीं हो रहा है तो कितना हो रहा है, and at what point what should be the next step, what should be the next thing that should be done for the patient. So I'll skip this. This is basically the various stages of development where how imaging can be used. Same thing, as you see, this is now brain. FDG, It's commonly used PET scanning because it can penetrate and it can tell you. Now, for example, in our center, my radiochemists are designing various chemicals for GBM, for researchers, for physicians, or for scientists who are interested in GBM, glioblastoma, brain tumor, to study that those radionuclide tagged chemicals which can help them to see. But now as a chemist thing, it's not just inside the body will light up karega. You have to have a chemical that actually also crosses the blood-brain barrier, right? So it gives you a lot of creativity to a chemist to design a molecule which has the property to be effective but also be able to cross the blood-brain barrier so that physicians, surgeons, scientists can actually see what is actually happening. So it is FDG, PET, commonly used in neurodegenerative diseases. Now, imaging is also used as a surrogate marker. When you can mark something that you're interested in by putting an imaging agent, you can use that to see kahan आपका drug जाएगा. So it's a surrogate marker for that. And you can just follow the image, body के अंदर किस organ के अंदर कहाँ जा रहा है, कितना जा रहा है, क्यों जा रहा है, जा पा रहा है, नहीं पा रहा, without actually having to do the treatment. You can, this is just a surrogate marker. So many applications, this just being one of those. So this is a complicated, but you will have the copy of this, not to worry. But point being, as you can see here, from target expression from early biology to compound screening, which is the drug discovery, drug development part. This is whole drug development part, assessment of pharmacokinetics, again up to that. Her stage के ऊपर various imagings are used. Imaging tells you whether to go forward or not. Go, no go decision you can make. It's just a modality, but it's an extremely powerful modality. And then, of course, in the clinical, as I mentioned, because you can image the patient and you know whether it's working or not working. Okay. I actually have made multiple topics for today. So I am perfectly fine if we take a shorter break and come back and I can go over biological therapeutics, RNA therapy, cell therapy, gene therapy, immune therapy. So whatever length of break you want, 10, 15 minutes, I'm okay. Right. Question, do you want me to continue or you want to take a break? You can take 10 minutes break, so I'll pull up my other slide and then you can, and I'm sorry, आप इस समय चाय पीते होते हो, आपकी चाय आ गई है? I didn't know that, you can check that. So, brain के blood flow जो है, कुदरत ने ऐसे बनाया कि हर चीज़ नहीं जाती. There is actually a layer that doesn't cross us there. So, you know what the brain, do you know makeup of human anatomy, brain कैसे है? Brain जो है, actually जो brain है is sitting in the fluid. जो concussion आपको पता है ना, जो फुटबॉल में अभी ये पता नहीं ये कुछ बंद कर दिया है कि header जो है उसको बंद कर दिया. There was actually a movie. Concussion is the name of the movie. क्योंकि ये तो बिल्कुल ठीक है, आपने रग्बी वालों को देखा है, रग्बी खेलते हुए आपने देखा, they wear no guard. ये बंद है ना, ठीक है, फिर मेरे को जोक्स आए तो फिर वो रिकॉर्ड ना हो जाए. आपने रग्बी वालों को देखा है, they wear no gear, खुलेआम, उनके कान देखें, रग्बी वालों के कान नोट करना, ऐसे पिसे हुए होते हैं. रेस्ट ऑफ द बॉडी तो बलून्ड होती है, मसल्स होते हैं, बट उनके कान जो है दे हेड बट करते हैं बहुत, और उनको जो ब्रेन इंजरीज बहुत होती है. सेम वे अमेरिकन फुटबॉल जो है, नॉट द सॉकर, द अमेरिकन फुटबॉल, जो दे वेयर हेलमेट एंड ऑल दैट, दे वेयर द गियर, बट उसका जो इट्स एक है ऑफेंस साइड होती है, एक है द डिफेंस. डिफेंस वाले जो होते हैं उनका काम होता है ऑफेंस वालों को रोकना. 200 kg, 250 pounds, 300 pounds. और वो हेड बट बहुत करते हैं। तो मैं क्यों स्टोरी यहाँ से शुरू कर रहा हूँ, वेलकम टू ब्रेन का, कि ब्रेन जो है इज एक्चुअली कनेक्टेड, इज सिटिंग इनसाइड, जो आपका जो स्कल है उसके अंदर फ्लूइड है, फ्लूइड में ब्रेन ऐसे तैर रहा होता है, तैर रहा इन द सेंस इज इट्स नॉट फिक्स्ड, इट इज मोबाइल, इट इज मूविंग। जब हेड बट करते हैं होता है ना एकदम से चक्कर आ गया, क्योंकि वो अपना फ्लूइड जो है के अंदर वो थोड़ा सा हिल गया। तो कुदरत ने बनाया इस तरह से कि हर चीज उसमें नहीं जा सकती, देयर आर लेयर्स so that not everything crosses through. So that barrier is called blood brain barrier. उसके अंदर क्रॉस करने के लिए, now if something imaging has to be done for the brain part, if the drug has to be developed the brain part, that whatever that drug is has to cross through the barrier in order to get into the brain. That is called blood brain barrier. हम मूवीज तो देखे हैं, बॉलीवुड इज गुड, पंजाबी वुड इज गुड, बट व्हाट दोज काइंड ऑफ मूवी, कंकशन, वो पूरी उसने बदल के रूल्स बदल के रख दिए, बट हाउ मच money influence and how much they, okay, but that's entirely based on how concussion happens in the brain. So when you make that, not every chemical crosses through, so you have to design drugs which can pass through this blood brain. So uska jo hai log p value, again as a chemist, lipophilicity, log p value उसकी adjust करते हैं कि क्या उसकी log p value होनी चाहिए so it crosses through. That's one of the property. Then again, as I showed you, you are juggling many balls and you're keeping all those in the air. So that's one of those balls that you have to keep track of. I'm perfectly fine. जब तक आपकी चाय आती है, I can start my next topic. आ गई है? 2 minutes, okay. So far as I'm concerned, I'm perfectly fine. When I'm giving talk, you are eating. Seriously. From where I come from, legally we don't stop them. It's up to you. You can take your coffee, यहाँ बैठ जाइए, मैं I'll continue. शायद ग्लूकोज अंदर जाएगा तो असर भी जल्दी होगा, ज़्यादा होगा. I'm just kidding. Just whatever you like, I'm fine. You can take your coffee or samosa and I will continue my lecture. which are currently, and I'll briefly go over these, RNA therapeutics, antibody based, which could be pure antibody or antibody drug conjugate, or cell based, which is CAR T, for example, chimeric antigen T cell receptors. So the field has evolved from, it's not that it's a new thing. पहले भी जो है, चाहे उसका नाम नहीं था biological therapeutics, but people used to use, for example, आप दही जो खाते हैं वो क्या है? It's a bacteria, right? It's a bacteria, उसका therapeutic effect है. तो same way things have developed. ये देखो, ये just one of the things that ever, इजिप्ट के अंदर मेडिकल हर्बल सिस्टम यूज होते थे, इंडिया के अंदर आयुर्वेदिक मेडिसिन, उसके अंदर जैसे एज आई मेंशन कर्ड, दही जो है, बट दीज आर माइक्रो ऑर्गेनिज्म, सो इट्स अ बेसिकली biological therapy, right? So these have been around for a while, and different places, different things at different timeline, as you see here on the screen, have evolved and have been used effectively. More recently, as we were talking yesterday, vaccine, RNA vaccine, COVID happened, and now there are so many, therapies based on COVID. So I'll quickly go over. So this is basically now, as more and more understanding of the biology, specifically of the immunology, the immune system, body का जो defense mechanism है, जो immune system है, उसकी understanding जैसे ज़्यादा होती जा रही है, people are trying to come up with the body's own immune system to fight against diseases. And that's basically simply what immune therapy is. And there are, so just if you look at the cartoon, traditional method जो है, immune therapy versus cancer therapy, which is जो अब तक के, what I've been telling you is drugs बनाने, drug design करने, but these are chemotherapy design which are from outside. Immune therapy, as I mentioned, which is uses body's own immune system to fight against the disease. And if you look at in terms of the scale at which size-wise, you look at the small molecules, nucleic acid bigger, peptides bigger, enzyme, antibodies commonly used, and cell-based therapies, microbiome. How many of you have read chemistry articles in microbiota? Do you know what is microbiota? आपका जो stomach है, everybody has their own microbiota. And that microbiota reacts, therefore different people will react differently to what next goes in, whether it's a therapy, ya khana. If you give, for example, people say they are allergic to something. Allergy, there could be many other reasons, but microbiota जो है, gut microbiome जो है, is different. A vegetarian versus non-vegetarian, उसका अपना genetic makeup कैसा है, कितना खाने की आदत है, वो सिर्फ रोज खाता है, सिर्फ मंगलवार को ही छोड़ता है या बिल्कुल नहीं खाता. All of those things जो है play these things. So that microbiota जो है is the reason I suddenly realized the microbiota and chemistry. Again, those of you all, those who are chemists interested in, it's a field which is slowly starting to open. And again, like I was telling, radionuclide, radiopharmaceuticals के अंदर जाने का, माइक्रोबायोटा के अंदर आप केमिस्ट बन के केमिस्ट्री की डिग्री यहाँ से ले, जहाँ मर्जी से ले, अच्छी तरह से मेहनत मशक्क़त करके आप माइक्रोबायोटा के रिसर्च में आप एंटर कीजिए. इट्स अ स्लोली ओपनिंग फील्ड, एंड दैट्स बेसिकली व्हाट इज हैपनिंग. सो ऑन द स्केल ऑफ एटॉमिक स्केल, मॉलिक्यूलर स्केल, small molecule छोटे से हैं, you can draw the structure, you can care. Then peptide, nucleic acid peptide, you can do that. But when you get into enzymes and antibody, they are larger. Now, larger, their properties could be different. Again, going back to the question asked about BBB, blood-brain barrier, how will they cross, or how to tag those. So, various of these cell-based, you give the whole cell as a treatment. So, cell-based therapy, if you are reading the latest literature, or if you have been reading literature, the gene therapy, cell-based therapy, these are the latest advancement. कि cells अपनी body के cells ही दिए जाते हैं against to fight your own disease. So if you look at, so as I mentioned, RNA, I will touch upon RNA, antisense, antibodies, and cell-based CAR T cells. So field on the human scale, if you think that it's ज़्यादा पुरानी नहीं है, 60s में ही उसका systematically बनना शुरू हुआ, and now 2018 there are RNA drugs, approved drugs, which are in the market. So how does RNA therapy works? So antisense, these are smaller makeup of RNA which can be designed to systematically go and interfere in the part of the body. These are small RNA sequence which are antisense part known. वो आपको design कर सकते हैं, that will go and tag into the protein of interest, and they do same biological intervention, stop the function of proteins that you don't want. जैसे overexpression of certain proteins leads to cancer, and that cancer basically those proteins then activate cell growth faster. Therefore, cancer grows, cells goes faster than the normal cells, right? If you can design an antisense, if you can design RNA, we can go and block that interfere into the progress in the repetition of the proliferation of the cells, that becomes a drug, right? So that's what basically what it does. Again, because they are smaller, they are also used as RNA delivery system. Where was drug delivery? कि अभी मैं बात कर रहा था drug delivery. This is drug delivery. And drug delivery, you can design, RNAs are used to again design to go inside the body and carry the cargo, quote unquote, your drug, which is the cargo, that RNA is the delivery boy or girl, which takes the cargo to the site of entry. So RNA based, there are many therapies which are based on the RNA and RNA, and here are a few like antisense. You know, these are the which are currently being developed, being used. There are drugs which are in the market based on RNA. So this is, you know, whether they are antisense, iRNA, interference RNA, like mRNA, messenger RNAs. You might have heard this, aptamers. These are all mRNA vaccines. अभी COVID से थोड़ी देर पहले छुटकारा मिला नहीं है. It has been diluted. The strain has been diluted. It is very likely, I think in the Western world it's already happening. It is becoming more like आपका flu vaccines जो होती है ना annually लेनी पड़ती है flu season के अंदर. उसी तरह से COVID का भी है कि it has become, because the strain is diluted now. So you probably take, because if you take the COVID vaccine, first, second, its effect is lasting only to some extent, and again, दूसरी strain आ जाती है. But it has been diluted and there are strains. So it will probably, I don't know how it's here, but हमें वहाँ पे जो है already instructions आनी शुरू हो गई कि this is more like you take annually, COVID vaccine जो है. So again, what is that? It's an RNA based vaccine जो है, or you can synthetically you design जो है RNA, or CRISPR-Cas9. Okay, I will leave that for you to study. But then again, it's one of those. So the advantage is that if you take an RNA-based vaccine, it will bind to the RNA of interest that you want to. It will cause a degradation faster compared to a small molecule which has to go and has to do that. So another advantage is that it's the ease, आसान है just to integrate with the existing therapies के साथ में. It's not too complicated. You can just mix and match. You can give multiple combination therapies with that. They are a bit unstable, and sometimes it could be a disadvantage depending on what you're looking for. The question for that is, if you have an RNA, what is the right target of that RNA जिसको आप target कर सकें? So that takes a little bit of research to be done. But overall, once you have figured it out, okay, which is the target for the RNA, you can design it's easier to develop as a therapeutics, relatively easier to develop as a therapeutic compared to a small molecule. लो अब ढूंढते रहो इस ओशन के अंदर को कहाँ जाके टकराएगा या कितना असर करेगा या किसी और को साथ में जाके वो टॉक्सिक इफेक्ट तो नहीं कर देगा. So there are advantages and disadvantages, and I will skip this because this is in writing and you will have it. What are the properties of antisense RNA? I was just, when I was preparing my lecture, I just thought probably I will include this. You don't need to go through this, you will have this in your handouts. And also, इसमें 25 पेपर हैं, review articles, which will related to all the lectures that I have given and the lecture I will give. So you will have, in addition to these, you will have a lot more information that you can just use as a reference. As you can see, it's nearly a book that I created, and you will have that. And I'll give it to Dr. Singh, and you will have all that information. So I'm not going to एक एक चीज पढ़ने वाला नहीं हूँ. But there is no point. I mean, you're not into kindergartners. You can just do some of this your own. So anyway, again, so I will not. So that's one technique, RNA-based technique, which is antisense, जो design कर सकते हैं आप RNA, smaller version of the RNA, and then you look for उसका target कौन सा है. It is very specific. Once you know what the target is, it will not interfere with anything. It will just go and interact with that. And if you can find that match, voila, it becomes highly, highly effective therapy. So it's a rapidly growing mechanism. And again, so the mechanism through which it works is find where it binds to, which RNA piece that it binds to, where it cleaves, where cleaving, and if there is a translational, translation and transcription, जो आपको बताया था, S phase, G1 phase, M phase, G2 phase, जो transcription जो हो रहा है, cell replication का, in the proliferation process, specifically if you know it can go and bind, design, it can help you to understand the mechanism part of this. So, and then if you can break that, क्योंकि ट्रांसक्रिप्शन में क्या हो रहा है, आपका पुराना सेल से नया सेल बन रहा है, राइट. एस फेज में स्टार्ट फेज है, गोज टू G1 फेज, माइटोसिस फेज, G2 फेज, उसमें जो आपकी जो इंफॉर्मेशन वो ट्रांसफ़र हो रही है ना, अगर बीच में ही उसको रोक दिया, अब आप सोचिए अगर कैंसर सेल है, वो ट्रांसफ़र होने जा रहा है, उसको बीच में ही ब्लॉक कर दिया तो आगे वो नहीं बढ़ेगा, तो देन जिसने उसको रोका वो ड्रग बन गया उसके लिए. सो दैट इज़ हाउ दिस एंटीसेंस और RNA बेस्ड थेरेपीज आर एक्सट्रीमली effective. Again, this is again I just put the list of various applications that you can have. What steps are taken, and the application part, a lot of application. Where disease, I said NCDs for example, non-communicable diseases, cancer, neurodegeneration, viral. We just now saw viral COVID के साथ में जो अभी देखने को मिला है कि जो वायरस को करने के लिए genetic disorder, there is a mental disorder, there these have been developed, are being developed, and there are many other rare diseases. So Ionis, IONIS is one of the company which is one of the leader in they have about 30 plus drugs in the market, specifically for rare diseases, because the diseases where there are many patients, pharma company उसमें invest करती है क्योंकि they have to answer to their stakeholders, they have to make money in order to continue to do that. जो rare disease है जिसमें patient ही बहुत कम है, there are not too many people who are interested in developing their science, क्योंकि अगर वो $1 billion invest करते हैं कुछ वापस आएगा नहीं. But there are companies who have mastered these techniques, and that's the truth. So Ionis is one of those company which has been since 80s. And RNA का, just as I was telling, there are antisense oligonucleotide ASOs, interference RNA, RNAis, CRISPR-Cas9 combination है. And again, I will not blabber on this. I already told you there are various, for delivery, for specific interaction, to get the immune response, but it's cost depending on how easily you can make or target. I will not touch cost on this part right now because cost is a business aspect. That's a different part. Now, आप लोगों ने नाम सुना होगा CAR-T, सुना है? It's a cell based therapy. आपने कार का नाम तो सुना है, वो तो सुना है ना, CAR-T are chimeric antigen receptor टी सेल्स इम्यून सिस्टम के अंदर। तो जो कार है उसके आगे टाटा लगा दीजिए, it becomes CAR-T. And that is one of the fast growing and one of the best way immunologists been working for last 30 years, 30, 40 years or so, कि अपने ही immune cells जो हैं उनको किस तरह से एक्टिवेट किया जाए कि वो डिजीज सेल के ऊपर अटैक करें। इट्स अ वेरी क्रिएटिव वे, वेरी स्मार्ट वे। यू हैव टू अंडरस्टैंड द होल थिंग कि अपने इम्यून सिस्टम को किस तरह से किया जाता है, उसके एडवांटेज एंड डिसएडवांटेज हैं। क्योंकि आप क्या करते हैं, आप इसके अंदर एनीवे, बेसिकली स्टेप्स इन्वॉल्व। इफ यू से you harvest immune cells from individuals peripheral blood, PBMC peripheral blood जो है उसमें से आप उसके ही immune cells को निकालते हैं। You harvest, these are the steps involved. You genetically engineer those cells, right? And you take those cells, so this is basically specific to where the target is. You create chimeric antigen receptor. These are T cells. Okay, आपने T cell, B cell का सुना है? Right? These are T cells. Tata है और bye bye है. Easy to remember. T cells and B cells are important immune cells in the body, right? So उस तरह से, so just think how creative way कि अपनी ही बॉडी से पेशेंट के जो हेल्दी सेल्स हैं उनको निकाला, उनको इंजीनियर किया और बाद में उसी को ट्रीटमेंट करने के दिया, सो दैट दे गो इनसाइड द कैंसर सेल और उसके ऊपर अटैक करें। वेरी क्रिएटिव, राइट? अभी जो है सेल बेस्ड थेरेपी पे जो सरफेस मार्कर है CD19 सरफेस मार्कर है, उसके करने के लिए इट टुक many years and eventually it worked. Immunologists worked for 30-40 years, it worked, and there is drug now, there are drugs now in the market. 2022, 21 के अंदर Jim Allison from MD Anderson, Texas, won the Nobel Prize for that cell-based therapy, for that therapy where it's for the immune therapy. And there is actually a, I think it should probably be online if you like, कि कितनी मशक्कत and कितना creative science करने के बाद how many people he had to convince that eventually working with the big pharma and show that it will work, and it worked, and it worked. So there is actually a documentary. I think many of these things when you Google you will find, and इनकी जो है जिम एलिसन की अपनी मतलब movies हैं, I have seen the movies on PBS के अंदर, Public Broadcasting System US के अंदर, you can Google and you will find it. But you see, the reason I'm mentioning is not कि movie देखो, I will not promote that, but scientific movie देखो, no, see, but किस तरह से how the field was developed, the whole जो जिसके लिए immune therapy के लिए उनको पता. But again, this is how it is done. You take the cells, collect the cells, you transfect the T cells, you engineer the T cells. After that, you give the T cells back to the patient, specifically उसको engineer करके कि किस specific कौन से target के लिए उसे करना है, you give back the T cells. जहाँ वो करता है, it completely wipes the cancer away. But challenge अभी यह है कि अभी it's in a developing field, क्योंकि एक बार आपने engineer किया, तो it starts sometimes इतना स्पीड से बढ़ने लगता है, तो जो काइनोम स्टॉर्म जो है कोविड-19 का भी एक यही वन ऑफ द रीजन था कि इतनी फटाफट वो फैलते हैं, जो आपको काइनेजेस जो है कीनोम जो स्टॉर्म जो है इतनी फटाफट फैलता था, तो उसके कारण इतनी टॉक्सिसिटी बॉडी में हो जाती थी, क्योंकि नॉर्मल फंक्शन इज वट एवर द सेल्स आर वर्किंग, वट प्रोटीन्स आर वर्किंग, बट किसी की एकदम इतनी ज़्यादा से होने लगी, ऑब्वियसली वो दूसरों को अफेक्ट करेगी। आसपास 1000 प्रोटीन्स है, हर प्रोटीन का अपना फंक्शन है, वो फंक्शन ऊपर नीचे होने लगता है, एंड बॉडी गोज haywire. So that control the toxicity of that has been a challenge, but things have evolved quite a bit now. So there are at least 5 drugs, CAR-T based drugs, which are in the market, and one of the leading person is from UPenn, Dr. Carl June. Again, I'm giving you these names, just read. Carl June, who actually mastered one of these techniques. So see, basically study started about 50 years ago कि bone अपना जो है bone marrow जो है उसमें से stem cells को निकाल के to study how those they behave, they work, what their functions are, and slowly piece by piece. Now this is again the work of thousands and thousands of scientists, biologists, immunologists around the world in order to understand कि machinery काम कैसे करती है, and then slowly how you can tailor it to make it therapeutically useful. Anyway, so as you see here, this started, first trial was in pediatric बच्चों के अंदर, because there are not too many drugs available for in the pediatric population, and 2013 में पहली थेरेपी गई थी, FDA ने 2014 में breakthrough therapy. Again, breakthrough and these things are the timeline with speed with which FDA can give you the approval to be used. But anyway, now there are multiple drugs, at least there are 5 drugs which are in the market. So if you look at immune therapy के लिहाज से, there are, you know, immune therapy very targeted. You can have biospecifics which are working there. You can have antibody. I'll come to you. I'll show you. CAR-T I just mentioned. I'll show you briefly about biospecific antibodies and antibody drug conjugate. Traditionally, radiation chemotherapy. I've been telling you about chemotherapy. Radiation जो है दिया जाता है, radiation, you know, radiation treatment दिया जाती है आपके. I mean, PGI also has departments of radiation oncology, for example. Stem cell transplantation, another methods which are used. So basically, ये जो machinery है, as you see, I've been telling you, machinery जो है, it's a very sophisticated machinery ever made is human body. And so there are obviously there are advantages, there are disadvantages. I have listed so some of these. The advantage is once it works, उसकी जो therapeutic effect है, जब उसने मार दिया जो enemies हैं उन cells को मार दिया, उसका effect is lifelong. इस तरह से नहीं कि बार बार लेनी पड़े medicine, बार बार आपको pills लेने पड़े, IV लेना पड़े, उसका नहीं है. If it works, it completely removes, wipes out the cancer cells. Lifelong उसका effect होता है. So those are again, you know, the so it is not just it's a therapy, it actually boosts the immune system. So it's more than just the therapeutic treatment. It makes your immune system, आपका जो defense mechanism है body का, उसको strong कर देता है. Advantage है. One of the disadvantages, for example, is it's a very expensive and fully not understood कि आपने जो बनाया है बॉडी के अंदर जाके कितना अटैक करेगा, कैसा अटैक करेगा, करेगा या नहीं करेगा, या उसका टॉक्सिक इफेक्ट होगा. सो इट इज़ नॉट फुल्ली अंडरस्टूड येट. जिन केसेस में, एंड मोस्ट ऑफ दीज केसेस आर जो है आइदर पीडियाट्रिक केसेस हैं, उनमें अप्रूव हुआ है अब तक. एंड देयर इज रिसर्च गोइंग ऑन अराउंड द वर्ल्ड इन बेस्ट ऑफ द लैब्स ट्राइंग टू फिगर आउट कि हाउ दिस इम्यून थेरेपी वर्क्स. So this is as I was saying, the Kinome syndrome. This is cytokine release syndrome. जो एकदम से तूफान एकदम से आता है, cytokines जो हैं एकदम से बड़े एक्टिव हो जाते हैं. अब इतने एक्टिव हो गए तो बॉडी की सारी दूसरी फंक्शंस है, they get affected. Heart, lung, brain, सब पे असर होना शुरू होता है. Eventually body cannot cope up with that. So that is the toxic effect. Understanding that is still will take some time to understand how that thing actually works. But where it works beautifully. And the other thing is, the other approach is antibody based. Antibodies are used as therapeutics. So you can see, for example, here, आपके जो antibodies हैं, Y shape में, उसका structure रहा, for example, आप antibody को tag कर सकते हैं अपने किसी भी जो टारगेट के अंदर आपने डालना है, और specifically उस टारगेट के अगेंस्ट आप antibody का वो है मिसाइल लगा सकते हैं उसको. So those techniques, there are many antibody which have been approved, therapeutics, which are mostly monoclonal antibodies, जो कि है, they work beautifully. Same way biospecific. This is basically two different types of cells, bringing those cells together. You take the antibody, as you see the Y shape, एक पे एक से उसको अटैक किया, दूसरे पे दूसरे से अटैक किया. So you take two different cells, bring them together biospecifically, so they can go and inside the body they can, two drugs or two functions can be done simultaneously. So those are biospecific antibodies which can be designed, which people design in order to understand the disease and or to for the treatment purposes. So yeah, and most often these are given as IV. Plasma में सीधा blood cell में IV source से दिया जाता है, so it can circulate quickly. They don't sit around in the body for too long. This is just an additional part of this, that you can design various monoclonal antibodies. You can put various biospecific of interest so that though different targets go inside. Remember, inside the body there are thousands and thousands of proteins. There could be multiple targets. So एक ही के अंदर आपने एक चीज़ को टारगेट के लगाया, दूसरे को दूसरी चीज़ पर टारगेट पर लगाया. Presumably you know that what those targets are. You have enough information, and then with that antibody you give the IV, it goes inside and it does its functions. It interferes in that the driver protein of your disease, and it can stop the disease. So that's how these are used. This is just the summary part, and you'll have this slide, not to worry. But this was just to show for you to understand and to appreciate that there are a lot of antibodies which are being based research that is happening. The antibody sources could be from different sources. Rabbit से antibody बनाई जाती हैं क्योंकि antibody you're just simply growing safer antibody and then you're putting tag क्या लगाना है. So antibody design, again, huge field. Now just imagine I mentioned antibody monoclonal antibody पे लगाना है. Now imagine कि आपका जो वही Y shape एंटीबॉडी का स्ट्रक्चर है उसके ऊपर आपने अपना स्मॉल मॉलिक्यूल जो कीमोथेरेपी है वो लगा दी, उस पर अटैच कर दिया, राइट. सो एक ड्रग, एक एंटीबॉडी which also has a therapeutic application and a drug which also has a therapeutic application. So you can take two drugs, and if there are the protein structure that you know and you want to attack on two different sides of the protein, now your missile actually has two drugs, एक तो antibody itself and then the small molecule, it going and attacking on that protein which is the driver, attacking that protein. So it's a growing field now, and there are I think about 19 drugs approved which is known as antibody drug conjugate. These are basically conjugate of antibody and the small molecule of interest. And there's tons and tons of research, and we are also doing some studies in our lab where same way, you take the drug, as I show here, have an antibody, you put one drug, you can put another drug, two different drugs of your interest. So in that case, antibody works as a delivery system because it's a warhead that takes the two drugs, two missiles, and attacks on the target. So you see here this cartoon that it shows, ये antibody आके cells पे लग गई क्योंकि it will bind to the surface protein, and now there are two missiles ready to interfere. So it can have a faster way. There, as I said, it started not too long ago, and there are already 18 or 19, I think, approved drugs in the market. So that's another area of interest. And basically, this is just again magnified picture of showing how you can your antibody drug conjugate जो है, that conjugate of various drugs on the antibody can be brought together, but it's a very targeted biopharmaceutical drug because it's very targeted because your antibody is specifically designed for a target. So it's very targeted, and then on top of that you can have a small molecule which is attached to it, which is also already a drug. So you have two drugs or three at least. Okay, that was the second drug. पहले ने बंद किया, दूसरे ने शुरू किया. Anyway, so that's basically another way, and I will not go into the mechanism part because that's basically it's more of the biology side. But as in a simplified, बहुत ही simplified way में, as I mentioned, you have your delivery system or a missile load, and you have two missiles which are attaching to the surface of the protein, and one is interfering in the one part of the another protein, उसके function को रोक देगा, तो cell growth को stop कर देगा. It could be the other one. So दो तरीके से आप उसको attack कर सकते हैं. The protein which is the cause of your disease. Okay. Any question? No question. I have third topic and I'm going to change the topic. Okay. Based on that, you design so it can crosses the blood-brain barrier. Okay. Based on that, you design so it can crosses the blood-brain barrier. So there are actually many assays which have been developed which tells you whether your drug passes through the blood-brain barrier or not. If right now, for example, in my lab, I have a postdoc from Germany who's been working for the last 3+ years on multiple this drug development project. For past year and a half, we've been designing drugs for one of the stroke function, brain function ke liye. And the goal is it has to cross. So he's designing, he's a chemist, he came as a simple, hardcore, synthetic, heterocyclic metallic chemist. उसने PhD खत्म की थी. Now after joining my lab, he talks only medicinal chemistry and proteins and how to target. He educated himself in computational modeling. He took online courses अपने आप बैठ के how computational modeling is done, how the chemoinformatics, all of these things, what programs download करके. Now what he does is that's what he does. अगर blood brain barrier को cross करना है तो what are the molecule structure, what are the features that should be, so that's what he does. He now designs different structure and come up with them. Then we have our biology actually in the cardiovascular department. We have one of my colleague who has another postdoc, came from another country, who's actually he's an MD, he's a trained physician, but now he's doing all lab research, trying to develop an assay for blood-brain barrier. We have a third scientist who came from UK. She does only develop these kind of assays which can cross the blood-brain barrier. So before you take it to animal, there are in vitro assays which are available, which can give you go no-go decision. So you follow those assays. Based off those assays, you narrow down. So if, for example, if he made 100, I think for now he has made about 150 new molecules, then he passes through एक साइंटिस्ट मेरी लैब में वो चेक करता है कि एंटी whatever disease के लिए है या नहीं. दूसरा चेक करता है कि उसमें अर्ग टॉक्सिसिटी तो नहीं है, वो कार्डियो टॉक्सिसिटी उसमें नहीं है. I have a third person who simply tests कि इसमें efficacy है, वो efficacious है या नहीं, right? Then it goes to other people who test कि whether it will go through the blood-brain barrier or not. So based on those multiple, and sometimes we use one of the company here. Once we are done, I mean, you can use any CRO, contract research organization. So one of the company here, for example, Jubilant, we use here. Eurofin is another company there. We give them some of our compounds once we have not done, because we crank many drugs quickly. जब उस stage पे पहुंच गया, we just let these guys just tell us this, whether it will go. But there are, आपका question कि there are assays. Those are in vitro assays. Once all those in vitro assays करने के बाद में जब आपको कुछ top candidate निकले, then now we are starting. As I am here, people and my colleague who is also cardiovascular scientist, he's in the cardiovascular department, Professor Abo, he's traveling somewhere else. But our scientists in the lab are now narrowing down all that information to see कि अब animal work किस में करना, what are the top candidates, कैसे उसको आगे लेके जाना. क्योंकि अब हमारे पास में information है कि whether the drug is efficacious or not, solubility is good, bioavailability of the drug is good, it passes through the blood-brain barriers. So we'll take those candidates and now we'll start animal studies. So that's how it's done. Most likely, अभी पता नहीं है, research बहुत चल रहा है. Research बहुत चल रहा है कि antibody, the question है कि antibody is a large molecule. If I just showed you, look at on this scale. Small molecule is 300, 400, 500 molecular weight. Think of this. So it's a large molecule. To cross the blood-brain barrier is not going to be easy, right? So there is still people are trying to design, okay, maybe there is something that they can find. There are many types. We are working on, for example, my lab has been working on some of the proteins. One of those are for prostate cancer. And the person who did initially studied that protein, University of Michigan, he was interested in developing peptide. We were interested in developing small molecule. His peptide was working in vitro very nicely. But when he took in vivo, hamara small molecule was working better because it can penetrate. That's the advantage. Peptide cannot, in those conditions, it cannot. So these are part of the, it's part of research. That's why जहाँ obstacles है, you try to overcome those obstacles. So that's, अभी पता नहीं है. There is tons of research happening across the world. Who knows? There might be something that may come up. But point being, इंसान है, शरीर है, दर्द होता है, आप लोगों ने दवाई निकालनी है उसकी. Is that good enough for motivation? I'm not a motivational lecturer, I'm a simple professor. But if that will motivate you, that will be a good thing. Okay, I'm going to change the gear and I have another topic to talk about clinical trials. Because tomorrow is my last lecture, I will be showing you combination of all the things that we have done to share a case study. कि मेरी lab में हमने किस तरह से किया. Yeah, I'll pick up. Tonight I will design something and I'll share with you guys. Clinical. So when we say experimental therapeutics, it's not only कि experimental word अगर आया है तो सिर्फ lab में ही है. Clinic में भी experiment ही हो रहे हैं जब तक कि वो actual drug नहीं बना है, right? So clinical experiments, clinical development is an extremely important part. बगैर उसके drug market में आएगा भी नहीं. So how these clinical trials are done? And I showed you this thing yesterday, for example, right? जो different phase है. Phase 1 is the exploratory drug. एक बार सारे preclinical studies हो गए, अब उसका first phase के अंदर in small set of healthy individuals, you simply give them to see that there is no toxicity before even you test the efficacy कि वो ड्रग काम करता है या नहीं करता. उससे पहले देखना है कि ड्रग मारता तो नहीं है. It's important, right? Because अगर वो करता है तो क्या फ़ायदा उसका जब तक कि अगर उसके साइड इफेक्ट रहते हैं. So initial test is done in a small number of patients to see. There are many healthy volunteers. So there are many. There are Western world में कई चीज़ें हैं. People don't hesitate to volunteer. अगर humanity को फ़ायदा होता है तो they come, they volunteer. There are people, इतने volunteers hospital में काम करते हैं, you'll be surprised. आपको ऐसा लगेगा कि hospital is run partly by volunteers. No cost, they don't expect anything. That's what their way of doing seva, giving back to humanity. There is a lot. हमें लगता है ना कि हम ही दान पुण्य कर रहे हैं. Come and see. Come and see. एक controversial बात बोलूँ. अपने बच्चों के लिए पैसा नहीं छोड़ते ज़्यादा. वो हॉस्पिटल्स में देके जाते हैं, वो यूनिवर्सिटीज़ में देके जाते हैं. यूनिवर्सिटीज़ में अगर मशीनें चाहिए उसके लिए देते हैं. मतलब नॉट के बच्चों को भूखा मारेंगे. इनफ, बट यू रीड इन द टॉप बिलियनर्स जो हैं, टॉप, दे विल गिव इनफ दैट देयर किड्स सर्वाइव एंड डू सर्वाइव इन द सेंस डू वेल. बट देयर आर मैनी, वो अपनी सारी वेल्थ जो है, आप देखिए जितने हॉस्पिटल जो कैंसर सेंटर और बड़े बड़े इंस्टीट्यूट चल रहे हैं, वो देखिए ना उनके नाम लिख दिया सो एंड सो फंडेड. उसको देने की ज़रूरत नहीं है. दे गिव. अगर मशीन चाहिए तो ठीक है, आई विल गिव यू मशीन. वही मशीन जो है 10 करोड़ की आती है, दैट्स फाइन, जस्ट सेंड मी अ नोट. जो दे सकते हैं, इट्स वेरी कॉमन. दैट्स ऑल. ड्रग डिस्कवरी डजंट रन ऑन द गवर्नमेंट के पैसे से. वो तो छोटा सा है, उससे कुछ नहीं होता. इट इज़ रन्स ऑन लॉट ऑफ द प्राइवेट मनी. इट्स अ वेरी एक्सपेंसिव एंटरप्राइज, एज यू कैन सी. So going back to volunteers, healthy volunteers से मुझे ध्यान आया, volunteering charity is a lot in the Western world. वो बोलेंगे भी नहीं, दे देंगे, चेक लिख के दे जाएंगे कि ठीक है, मेरी ज़िंदगी मैंने जितनी थी जीनी जी ली थी, इतना है, थोड़ा बच्चों के लिए दे दो, आप अपने बच्चों को दे दो, और उनके स्कूल के अंदर बिल्डिंग बनानी है, बिल्डिंग के अंदर मशीनें लेनी है, NMR नहीं है, ठीक है, ले लो। सो वॉलंटियरिंग, अपनी हेल्थ को आगे लेके आना इज द फर्स्ट थिंग जहाँ पे कई चीज़ें टेस्ट होती हैं टू सी कि देयर इज़ नो टॉक्सिक इफेक्ट। एनीवे, फिलॉसफी ऑन वन साइड, practical thing on the other side. Okay, that's very common thing. Okay, because the idea is for a new drug to be effective, but before effective, it should not be toxic. If it is toxic, it defeats the purpose. Okay, so that's what phase 1 is done. It's a double-blinded. Do you know what that means? Patient को नहीं पता कि उसको क्या दिया जा रहा है, और जो एनालाइज कर रहा है उसको नहीं बताया जा रहा क्या जाता है। डबल ब्लाइंडेड, सिर्फ बीच में जो फिजिशियन, जो रिसर्चर है, उसी को पता है, सो दैट कोई इन्फ्लुएंस ना करे एक दूसरे को। विटामिन ए और कैंडिडेट ए इज गिवन एंड इफेक्ट बी इज ऑब्जर्व्ड, ना उसको पता है, ना जो एनालाइज कर रहा है उसको पता है। सो दे आर बोथ इंडिपेंडेंटली गिविंग देयर बेस्ट ऑब्जर्वेशन, बेस्ट इंटरप्रिटेशन। अब फिजिशियन विल कलेक्ट, साइंटिस्ट विल कलेक्ट टुगेदर एंड सी व्हाट इट मेक सेंस। आइडिया अगेन इज कि कोई एक दूसरे को इन्फ्लुएंस ना करे। सो दैट्स हाउ इट इज अ प्रोटोकॉल, इट्स अ रेगुलेटरी रिक्वायरमेंट, इट शुड बी डबल ब्लाइंडेड स्टडी। अगेन क्वेश्चन इज टू अंडरस्टैंड द सेफ्टी। सेफ्टी क्या है, टॉक्सिक तो नहीं है, सेफ्टी प्रोफाइल चेक करने के लिए इट इज गेन एंड अगेन ये छोटी छोटी चीज़ें, फूड इंटरैक्शंस, आप फूड ले रहे हैं, उसका कोई साइड इफेक्ट तो नहीं हो रहा ड्रग के साथ में, वो एब्जॉर्ब बराबर कर रहा है, उसका क्लियरेंस बराबर हो रहा है, ये इन चीज़ों को इफेक्ट करने के लिए। इस समय आप ये चेक नहीं करें कि उसका ड्रग इफेक्ट है कि नहीं। फेज 1 में उसकी दैट इज़ नॉट द टारगेट। फेज 1 इज सेफ्टी स्टडी एंड जस्ट फ्यू ऑफ दीज, एंड अगेन in specific population, as you see I have mentioned here. So age, gender, if there is a pre-existing condition, remember I was telling you if it's a diabetic, non-diabetic, just an example कि उसको कोई पहली pre-existing condition तो नहीं है, अगर वो condition है तो drug interaction तो नहीं हो रहा, ऐसी कोई चीज़ तो नहीं हो रही। So उन चीज़ों का ध्यान रखा जाता है कि कोई नया drug introduce करने से पहले वो drug का कहीं असर तो नहीं हो रहा। So that's base used. I mentioned about imaging yesterday. You can use imaging as to follow the drug, क्या उसका हो रहा है, but then again, not just the preclinical application, but imaging as you can see the application in the clinical side, कि उसका क्या असर होता है, कितना हो रहा है, वो उसका मेटाबॉलिज्म किस तरह से है, उसका क्लियरेंस, एलिमिनेशन प्रोसेस ठीक है, फार्माकोडायनामिक, उस ड्रग का असर कितना हो रहा है बॉडी के ऊपर। एक तो बॉडी फार्माकोकाइनेटिक्स, बॉडी क्या कर रही है, और ड्रग क्या कर रहा है, उसका असर कितना हो रहा है, उसका कोई अगेन हार्ट फंक्शंस पे तो कोई असर नहीं हो रहा, बिकॉज दैट्स अ टॉक्सिक इफेक्ट, राइट। सो सेफ्टी पैरामीटर्स जो हैं वो फेज 1 स्टडी में किए जाते हैं। दैट्स व्हाट फेज 1 स्टडीज आर फॉर। एंड अगेन जो एंड अगेन इट्स डोज फाइंडिंग स्टडीज अलग से की जाती है, वो भी अंडर फेज 1, कि क्या डोज ठीक है। आप किसी को 2 मेग पर किक देना है या 5 मेग पर किक देना है या 15 मेग पर किक देना है, कि कौन सा टॉलरेटेड डोज है, राइट। MTD क्या है, MTD आपने सुना है, मैक्सिमम टॉलरेबल डोज, कि जितना वो टॉलरेट उसको लेके जाते हैं कि ठीक है उस डोज पे है, उसका पीके अच्छा है, उसका प्रोफाइल अच्छा आ रहा है, तो ठीक है आप उसको इसके अंदर हम अब उसको बड़ी पेशेंट पॉपुलेशन फेज 2 वगैरह में हम लेके जा सकते हैं। सो initially phase 1 studies are for the safety, dose finding, looking at the PK/PD, उसका असर क्या हो रहा है, that it's done purely for that person, and to see there is no side effect. आप लोगों ने कितनों ने थैलिडमाइड का नाम सुना है, सबने सुना है, right? It changed the way, and again if you truly know the story of thalidomide, chemistry, purely chemistry, 2 isoform S and R, और बॉडी के अंदर जो है उसका ट्रांसफॉर्मेशन एक तो चेंज होता है, राइट? जापान अबाउट 10,000 crippled babies deaths happened. FDA में थी कैली यूएस के अंदर, शी स्टॉप्ड व्हेन शी वाज stringent के यूएस के अंदर वो ट्रायल ड्रग नहीं आएगा, एंड शी सेव्ड मेनी लाइव्स. बट थैलिडोमाइड इज अ केस स्टडी विच इज यूज्ड नाउ. थैलिडोमाइड इज नाउ बीइंग से लिप्रेसी के लिए फिर से ड्रग आ गया है. People have tested for leprosy, thalidomide का फॉर्म चेंज करके. Again, that's one of the reason कि उसका adverse drug effect क्या है उसको स्टडी करने के लिए. Does repurposing, for example, आपने एक ड्रग लिया, एक के लिए काम कर रहा है, you see that there is potential for that drug could be having application for something else. तो उसमें आपको पता तो चल गया because the drug has been already used in one form or the other for one or other application of one disease. Maybe it has additional application, so उससे आपका कम पड़ जाता है, but still you need to do. सेफ्टी उसमें हो चुकी है क्योंकि ड्रग इज बीइंग यूज्ड, सो इट्स सेफ. बट उसका डोज क्या है, कौन सा डोज कौन सी डिजीज के लिए वो एडजस्ट करना है, यू कैन डू दैट इज अगेन इन phase 1 के लिए. एंड देयर आर I just have, and you will have all of this. I have just given some sites यहाँ पे so that you can just on your own time you can just go through those sites, you can click and you can learn more about. So basically, what is clinical trial? Once you have shown that a drug is safe, now you want to see whether it's efficacious. जिस चीज़ के लिए उस ड्रग को बनाया गया उस डिजीज़ के लिए वो काम करता है या नहीं करता है. आपने बता दिया कि सेफ है, वो आपने डोज उसका एडजस्ट कर लिया, यू फाउंड आउट व्हाट द डोज इज द इफेक्ट, उसका जो डोज जो टॉलरेबल है जिसके ऊपर ट्रायल की सकती है. नाउ यू वांट टू शो इन ए लार्जर पॉपुलेशन कि वो ड्रग काम करता है. सो इट कुड बी phase 1 trial could be depending on क्या trial design और किस तरह से, it could be from 20 to 100 patients. Phase 2 trials could be from 200, 400, 500, 800 patients. अब देखने के लिए कि वो efficacious है या नहीं, वो काम उसका जो actual drug effect कितना है, medicinal effect उसका कितना है, और उसका interventional जो भी जिस चीज़ के लिए बनाया गया है वो उसका ट्रीटमेंट पे काम करता भी है नहीं करता है. So there are many steps that are involved, which are again, phase 1 as I mentioned, शुरुआत करने के लिए. Phase 2 and phase, the goal is phase 1 में जब safety हो गई उसकी, अब second उसकी efficacy को check करना है. So initial phase therefore is considered discovery phase. Initial phase, when phase 0, phase 1 is considered discovery phase, you are trying to still discover. उसके बाद में इस डेवलपमेंट स्टेज, क्लिनिकल डेवलपमेंट, और इट्स क्लिनिकल डेवलपमेंट when you have a candidate in a preclinical condition, right? You have identified the lead compound which works and you take it to animal studies, and in the animal study then you are trying to do more advanced studies. That's the preclinical development. एक बार क्लिनिक के अंदर करने के बाद when it enters the phase 2 trial, it's called clinical development. The terminology used, and phase 2, phase 3, because it's still not in the market. You're clinically trying to see phase. So once you reach to the point where when you have preclinical सारी studies कर दी हैं, animal studies हो गई, उसकी ADME, tox, safety सब हो गया, is safe. Now you want to go and try phase 1 trial. आपको रेगुलेटरी बॉडी के पास भी जाना पड़ेगा, जैसे यहाँ पे CSIR है इंडिया में है, नो सॉरी ICMR. यू गो देयर, US में जैसे FDA है. सो इट्स इन्वेस्टिगेशनल न्यू ड्रग्स, इट्स नॉट स्टिल अ ड्रग, इट्स एट द इन्वेस्टिगेशनल स्टेज. यू फाइल इतना बड़ा पोथा, यू हैव टू गिव एवरी डिटेल इंफॉर्मेशन अबाउट इसकी एफिकेसी, सेफ्टी, कितने स्पीसीज में क्या क्या डेटा आया था, किस तरह से आप लेके जाएंगे, क्या प्लान है आपका, क्या उसका प्रोटोकॉल होगा, सब कुछ देखे. सब 250, and that itself is a huge discipline, and it's a business field. They are experts, that's what they do. क्योंकि आपको पता भी होना चाहिए ना कि आप क्या कर रहे हैं. ड्रग की क्या रिक्वायरमेंट है, क्या होना चाहिए. It's a, just imagine they have to have so much knowledge about all aspects of this to put the packages together. Anyway, that's called IND, investigation new drug. You file that applications. Once you file the application, that's when the regulatory body will give you an approval कि ठीक है, वो सारा रिव्यू करते हैं कि आपका डेटा इज ऑल दैट इज फाइन. Go ahead and start the trial. That's when you enter the phase 1 trial, या phase 0 trial. So difference between phase 0 and phase 1. Phase 1, as I mentioned, safety trial है, can be done into up to 20 patients, so just to safe है या नहीं है, उसके अंदर कोई toxicity वगैरह नहीं है. Phase 0 is another protocol that was developed in other places and in various diseases, but in cancer, in oncology, it started about 2004, 2005, nearly 20 years ago. It came published in 2009, the first study, which is a phase 0, which means जो doses है, it is not to 20 patients, it is given to only 3 patients. Up to 3 patients, you see what happened. Based of that, it's a pharmacodynamics driven, not pharmacokinetics. कि drug आपका क्या असर कर रहा है. Based of that, you adjust in the next set of 3 उसके basis पे you go to the third. So between 6 to 9 patients, you know कि आपका drug का क्या असर है, होगा या नहीं होगा. Then you go to phase 1 trial. Like sometimes phase 1 efficacy के साथ में भी कर सकते हैं. It's called phase 1B, जिसमें आपने सेफ्टी टॉक्सिसिटी भी की है और उसके अंदर बढ़ा के उसके अंदर एफिकेसी भी उसकी चेक की है. दीज आर अगेन व्हेन यू गेट टू दैट, दीज आर ऑल रेगुलेटरी स्टेप्स दैट आर टेकन. सो फेज 2 बेसिकली क्या है, you take group of patients in the disease of interest for which drug has been developed, and that could be several hundred patients, and those are specifically in clinic because clinic में मिलेंगे आसानी से patient. Not you just try to do it in hospitals instead of going to private practices में सब जगह करने के लिए. आजकल, I mean, this is only having access to the patients. There are many approaches that are taken, but that's usually in a standard way. So again, goal is test the safety, at the same time start looking at the efficacy के patient काम करता है नहीं करता. That's the main thing. So imagine close to $1 billion इन्वेस्ट किए हैं, $500 million प्री क्लिनिकल पे इन्वेस्ट किया, फेज 1 शुरू किया है. उसके बावजूद only 1/3 of the drug realistically go forward, या कुछ ना कुछ तो उसमें आ ही जाता है. The drug does not go forward, उसमें the drug is not allowed to go forward. So just imagine pharma industry जिसने $500 million और so इन्वेस्ट किया है और वो ड्रग उसके 3 में से 2 ड्रग फेल हो गए, तो उन्होंने कॉस्ट कहीं से तो निकालनी है ना. दैट्स वाई ड्रग कॉस्ट आर एक्सपेंसिव, क्योंकि जो एक निकला उन्हीं में से उसने निकालना है बाकी की ट्रायल्स को आगे लेके जाने के लिए, बाकी की रिसर्च को आगे. एंड देन अगेन देयर इज ऑब्वियस प्रॉफिटेबिलिटी, शेयर होल्डर्स एंड ऑल ऑफ दैट. बट दैट्स रियली, if you, the number that I have, approximately 33, that means 1 in 3, nearly 1 in 3 is what actually gets to pass phase 2. अभी वो phase 3 में भी फिर जाना है उसने. And phase 3 basically is now patients of the disease being studied, now several hundred, and it could be smaller depending on the rare diseases, but more prevalent diseases जो हैं, and rare की definition also varies. Rare disease, what is the definition of rare disease again? And it could be worldwide, it could be anywhere. It could be through large hospitals, small hospitals, it could be private practices. The idea is now you have shown that the drug is safe, you have identified कि उसका dose, effective dose क्या है, it is efficacious in phase 2 trial, आपको पता था कि safe है और efficacious है. अब आप शो करना चाहते हैं कि मैंने उन गिने चुने 200 लोगों में किया है, उतना ही नहीं, अब आप वर्ल्ड वाइड बहुत सी डिफरेंट सेटिंग्स में, एनवायरनमेंट में आप उसको टेस्ट करके शो करना चाहते हैं. दिस यूजुअली few thousand people tested worldwide. That's basically people come, I mean patient come, phase 3 trial में they participate, they get the drug, and उसके अंदर उसकी efficacy आ रही है. Then you go with another dossier to FDA और या जो भी regulatory agency आपकी कंट्री की है, और सारा डेटा जो है आपका प्री क्लिनिकल से लेके क्लिनिकल सारा डेटा जाके उनके सामने रखते हैं कि देखिए हमने ये सब किया है, दिस इज द डेटा कि ड्रग इज एक्चुअली वर्किंग, इट इज सेफ, इट इज वर्किंग, एंड वी हैव ट्राइड इन मैनी डिफरेंट जियोग्राफीज, जेंडर, एज ग्रुप, हर चीज़ ट्राई किया, एंड ड्रग इज इफेक्टिव. That's when the companies get their drugs approved. Company या जो भी उसका applicant है gets the drug approved. That's when drug comes to the market. So just imagine it's an army of best of scientists from different disciplines get involved. अपने अपने एक एक स्टेप छोटा छोटा अपना एक एक बूंद लेके आके उस बकेट में डालने के लिए अपनी नॉलेज को. That's when, and then a huge investment. Then go to people trial, and in that, once you achieve the goal in phase 1, phase 2, phase 3 trial, and again 1/4 of the drugs will fail there also. In case of cancer, it is even stringent. About 95% of the drugs fail. Only 5 to 7% of the drug pass. It's even tough. The bar is even higher to achieve that. So, क्योंकि उसका जो है toxicity and all that has to be matched. And then clinical trials. Clinical trials again should be randomized. ऐसे नहीं कि आपने एक ही जीन के पूल के पकड़े और सिर्फ उन्हीं को टेस्ट किया, बोला जी मेरा ड्रग अप्रूव कर दो, मैं सारी दुनिया को देना चाहता हूँ. नो एजेंसी विल से ठीक है भाई, गोरा है, काला है, पीला है, नीला है, आदमी है, औरत है, बच्चा है, आप सब में करो क्योंकि आपका ड्रग तो मार्केट में जा रहा है, कोई भी यूज़ करेगा. सो दैट यू हैव टू शो दैट इट इज़ अ रैंडमाइज्ड trial. It is not just specifically target. If you have to do that, you can do that, but then has to be labeled accordingly that the drug is for only for that. But usually in a phase 3 trial, these are again randomized trial. Placebo, you know what is placebo, right? I'm assuming. Placebo trials, आपको ड्रग नहीं बाकी सब कुछ दे दिया, उसका कितना और पैरेलल में ड्रग दिया, तो आपका जो कोहॉर्ट जो होता है पेशेंट का, उसमें हेल्दी वॉलंटियर्स वगैरह और जो इसमें वॉलंटियर्स वगैरह आते हैं, उसमें सिर्फ प्लेसिबो दिया जाता है, नो ड्रग, और उसके पैरेलल जो कोहॉर्ट होता है पेशेंट का, उसके अंदर ड्रग दिया जाता है उसको, तो दोनों का कंपेयर किया जाता है. यू सी, आफ्टर ऑल आपने पब्लिक के लिए देना है तो आपको सारे स्टेप्स लेने जरूरी हैं कि कहीं तुक्का तो नहीं है, इज इट अ रियल थिंग. सो दैट्स व्हाई दिस इज अ वेरी tedious task. Blinded again. Blinded because you don't want it to be influenced. किसी भी तरह की influence नहीं होनी चाहिए. It should be very honest, neutral results which should be analyzed. So it has to be blinded so that there is no influence. And then there has to be an end point. आप क्या achieve करना चाहते हैं? आपकी इतनी efficacy show करता है, इतने टाइम में show करता है, आपको पहले से define करना पड़ता है कि यहाँ तक मैंने इतना achieve किया तो I would consider this as a success. और उतना नहीं हुआ तो you will. So these things are decided कि आपका गोल क्या है. आप पहले उसको बताइए कि आपका गोल क्या है. आप ड्रग लेके आप देखना चाहते हैं कि ड्रग जो है उसका असर होगा, आपका 70-80% जो है रिस्पांस रेट अगर उसमें आता है, 50% रिस्पांस रेट आता है, डिपेंडिंग ऑन व्हाट डिजीज इज, इतना रिस्पांस रेट आता है. आप पहले से डिसाइड करेंगे ट्राई करने से पहले. देन ड्रग गोज टू द मार्केट. अब अगर उसने आपने जो कहा था वो करेगा, अगर वो हुआ और आप वही कहेंगे जो आप पहले टेस्ट कर सके बेस्ड ऑन द डेटा. सो कंपनीज व्हेन दे डू दिस थिंग, दे डू एक्सटेंसिव अमाउंट ऑफ इन्वेस्टिगेशन स्टडी एंड देन दे सेट दैट क्राइटेरिया उसके अकॉर्डिंगली. एंड व्हेन द क्लिनिकल ट्रायल्स आर दे आर ऑलवेज नर्वस क्योंकि उन्होंने $2 billion तो आई मीन साइंस इज एक्सपेंसिव, इतना इन्वेस्ट किया और आखिर जाकर उनका अप्रूवल नहीं मिला. उनके तो $2 billion गए, नॉट जस्ट कि डॉलर गए, लोगों की इतनी मेहनत, इतनी आस, सब कुछ गई. सो दीज थिंग्स आर जस्ट इमेजिन कितनी वेरी टाइटली रेगुलेटेड स्टेप्स आर इन्वॉल्व in trying to develop a drug beyond preclinical part. So rare disease, same thing. I mean, if there are smaller number of patient population, ऐसी कोई disease है which is rare, there are not that many patient, उसका criteria थोड़ा different है. There are rare diseases, orphan diseases जो कि ज़्यादा नहीं है, कम है, उसके लिए criteria, but again steps are same, maybe less number of patient because patient अगर available नहीं है तो किस में test करेंगे. So those are again regulatory agencies and again इन clinical trials में कौन कौन involve होगा. Investigator ने तो एक बार पकड़ा दिया, investigator का काम हो गया. जिसने उसको own किया legally, जिसने own किया, sponsor, pharma company हो सकती है, university हो सकती है. Universities usually don't get involved because it's a very expensive process. Universities don't have that kind of money. Or universities are non-profit. Investigator जो जिसने मेहनत करके उसमें इन्वॉल्व हुआ है. एक्सक्यूज मी. सो स्पॉन्सर जो उसको स्पॉन्सर कर रहा है, इन्वेस्टिगेटर में जिसने मेहनत की है, coordinator who are coordinating, and of course the patient, the participants. So these are basically how you the sites. As I was telling, it should be randomized, it should give you the data which is useful ke different racial makeup में, different gender में, different age group में, ये सारा क्राइटेरिया के अनुसार ट्रायल्स सेटअप की जाती हैं. एंड देन ऑफ कोर्स देयर इज अ लीगल फ्रेमवर्क, क्योंकि रेगुलेटरी इन्वॉल्वमेंट होना जरूरी है. लेफ्ट एंड राइट आप नहीं दे सकते बगैर उसके बराबर किसने चेक्स एंड balances are not in place. And then based of that guidance डेवलप किए जाते हैं कि किस तरह से होनी चाहिए ट्रायल, कैसे उसकी स्टडी होनी चाहिए, कितनी होनी चाहिए, who will do the study and how the study will be done. All of these things are then designed accordingly. I mean, this is all just, and there are these FDA. I'm based on the US. I'm just, these are the things on paper what I've been telling you. And again, it's the responsibility of the sponsor company. जो भी उसको, उस sponsor means sponsor of the application to get approval. The responsibility उसकी है कि ट्रायल जो उसने लिख के दिया है कि इस तरह से हम ट्रायल करेंगे, strictly उसको follow करें. इसीलिए, as you know, FDA has, you probably may know, FDA has offices in many different countries. In every country जहाँ से ड्रग आते जाते हैं, टेस्ट होते हैं, FDA has offices there. Even in India, I think there are a dozen offices in India in different cities. I don't know if there is one in Chandigarh or not, but I know many parts of the country there are offices. Why FDA के offices यहाँ बैठे हैं कि clinical trials अगर कर रहे हैं, यहाँ से कुछ सामान ले रहे हैं, it is of quality or not. So worldwide, that's how the regulatory agencies they can have there. And I'll just skip this. So, and this is where you can get more information. With that, now it's your turn. Any question? Yes? To skip a phase? Why and phase 1 is for toxicity. कि वो टॉक्सिक है या नहीं. ठीक है, ड्रग इज नॉट टॉक्सिक. नो ना कि कौन सा फेज स्किप करेंगे. आइडिया इज वट एवर यू कॉल इट फेज 1, फेज 2, फेज 3, जो भी करना. आइडिया इज कि यू शो कि योर ड्रग इज सेफ, देयर इज नो टॉक्सिसिटी, देयर इज नो साइड इफेक्ट. यू हैव टू शो द ड्रग इज एफिकेशियस. हाँ, it is possible. I said you call whatever name you call. The fact is, in humans, you have to show that it is safe, no toxicity, it is efficacious. It actually has the effect for which the drug was developed. And phase 3, it works in a wide range of geographies, wide range of gender makeup, racial makeup. वाइट्स में काम करता है, इंडियंस में काम करता है, चाइनीज़ में नहीं करता है, अफ्रीकन अमेरिकन्स में नहीं करता है. ऐसे निकलता है तो देन यू नीड टू नो कि इट इज़ वेयर इट वर्क्स और वेयर डजंट वर्क. सो दैट रिगार्ड्स फेज 3. सो आप फेज 1 कहें और ये सारी चीज़ें करें, डजंट मैटर, यू कॉल वट एवर फेज यू वांट. बट ट्रेडिशनली दैट्स हाउ वी बिगिन. दैट्स अ गुड क्वेश्चन, क्योंकि 5-6 साल पहले 800 पेशेंट का मर्क ने एक ट्रायल की थी. फेज 1 के अंदर वो इट वाज सॉर्ट ऑफ अ वेरी इफेक्टिव ड्रग. फेज 1 के अंदर इंस्टेड ऑफ 20 पेशेंट दे कंटिन्यूड एंड डिड 800 पेशेंट एंड दे डिड ऑल दीज स्टडीज. तो फेज का नाम आप जो भी दे दें, एज लॉन्ग एज यू प्रूव दैट ऑल दीज थिंग्स आर वर्किंग, ड्रग इज सेफ, no side effect or manageable side effect, it is efficacious, it works in different geography, race, gender, आप वो सारा prove कर दें and FDA gave the approval. So you can skip in the sense, you can skip everything, but these things are needed. Phase का नाम कुछ भी आप दे दो. The way structure is yes. The way a regulatory structure has been set up, yes. You have to पहले में थोड़े में show करोगे it works, and then you take it to the larger and then take it to the larger population. COVID was the biggest trial. Billion people were involved. Before what? Sorry. Before, but they were trying. Nobody was listening to any regulatory. Everybody was there. India में अपनी trial हो रही थी, जो भी Indian companies हैं वो बना रही थी. हर country जहाँ पे भी drug development हो रहा है वो अपने अपना बना रहे थे और अपने लोगों में कर रहे थे. क्योंकि the enemy was unknown. पता नहीं कि वो COVID-19 है क्या, जब तक उसका समझ में आया. तो everybody was doing trial and they were sharing data. Agencies were there sharing data. Scientists were sharing data. So that actually, you know, we learned from past, so that actually has helped in changing many regulations. So vaccine का expedite है. I mean phase 1 का, like she asked, phase 1 का जो है approval of a drug in a phase 1. Phase is, now you know why phase का नाम दिया गया. पहले आप safety check कीजिए, फिर उसकी efficacy check कीजिए, फिर show कीजिए कि efficacy is efficacious in a larger setting, larger geographies world की. अफ्रीका है, इंडिया है, चाइना है, यूरोप है, ऑस्ट्रेलिया, जहाँ भी है. You show that. अब उस सारे को लाके आप कह दो phase 1 है, चलेगा. Data should have all of this information. Vaccines का भी lot of things are, we have learned a lot from that vaccine. COVID-19 has taught a lot. किस तरीके से, not in terms of what the research part, but in planning part. So there was an agency which was set up in the US, ARPA, which is specifically thinking of what might happen. COVID से सीख के, they started a few $1 billion investment करके के, what might happen. You start studying now. कि हमारे जो इतने कर्म ऐसे हैं कि कुछ भी, what might happen, so you start proactively doing, and the ARPA whole agency has been set up. But it's, we learn and then we change, we adjust accordingly. But so yeah, vaccine is teaching a lot. I mean infectious diseases research ऐसा लगता था कि जो सीखना था सब कुछ हो गया. कोविड ने आंखें खोल दी कि anything can happen. So you keep on actually learning. Human biology is the most sophisticated thing to learn. So when I do a program, चलो बंद है.
Welcome back everyone. We have Dr. Aman Bhalla back with us for the tutorial, which is a continuation of the morning session. The topic is asymmetric synthesis in drug design, auxiliaries and reactions, part 2. We look forward to more in-depth exploration of this essential topic in modern synthetic chemistry. Let's begin the session.
A very good afternoon. Okay. So in our morning session, we have studied about the asymmetric aldol reaction. Okay. So by using one auxiliary, we planned one enantiomer selective product. Okay. So now we have to move the last part of this asymmetric synthesis in drug designing. So again I am coming back to the reaction which we are talking in the morning regarding the asymmetric aldol reaction. So if seeing on this picture, just a one question has been asked to you, that how will you plan any one enantiomer out of the 4? Give answer in just 2 sentences. Without any mechanistic details, then how you are going to answer that? Without checking the mechanism or the detailed reaction steps, just on the basis of the theoretical concept which we discuss, can you answer for that? Okay. And the second question, if it is associated with it, out of these 4, how you classified them? You convert it into 2 groups. One is your syn enantiomer and the other is your anti enantiomers. But within this you can differentiate in syn both OH and R on the same side, or it can be on the opposite side. So now you have to think the answer of the question which I asked, and the answer is very simple in 2 lines. First you have to say that in this reaction which is on your screen, if you understand the 2 to 3 keywords of asymmetric synthesis, you can easily going to attempt or answer this in interview. The first question that you have to say that in the selection of E and Z enolate will lead to either of the group. Okay. So if you have a Z enolate, what type of enantiomer you are going to get? Syn enantiomer. What is Z? The groups are on the same side. What is E? The groups are on the different side, opposite side. So just by looking this picture, I said you don't know any mechanism. Okay, you don't know any steps. But if you listen 4 to 5 lectures very carefully, you have to just give the keywords, that how you are going to attain any one in majority. So you can say in first line, by appropriate selection of either E enolate or Z enolate, we can rule out the 50% of the product. For example, if you choose or plan or introduce Z enolate in your reaction, you will exclude anti enantiomer directly, because Z enolate is always going to give you syn. So if you choose E enolate, then you 50% Z syn enantiomer will be out of this. So by just choosing appropriate enolate, your 4 products are now possibly convert into only 2 product. Now come to only 2 product. For example, syn enantiomers. How you are differentiating between 1 and 2? In 1, OH and R on the above the plane, and in 2, OH and R on the below the plane. So if you want to attain number 1 molecule, you have to block the other face. If you have to attain number 2 molecule, you have to block the upper one. So this is the 2-liner answer if you know about the asymmetric synthesis. That in any chemical reaction, they can ask and they can give you this example and just give the answer if you know about asymmetry. They are not asking about any mechanism. Just looking into this, you can directly tell that how you are going to get one particular type of product. Okay. Now if you see this reaction, okay, if I go to this reaction, can you identify how these substrates are going to react to give the final product? In your first product, you have ACOH. Which functionality of this substrate is going to participate? कौन सी करेगी? प्रोकायरल कार्बन किसके पास है? कार्बोनाइल ग्रुप के पास. तो you have a 2 atom, carbon and oxygen. Now go to the part of your enolate. You have a carbon-carbon double bond, oxygen negative and metal positive. Now when these substrate are coming in this space, carbonyl के 2 atom, enolate के 4 atoms. enolate के क्या हो गए? कार्बन-कार्बन डबल बॉन्ड, ऑक्सीजन नेगेटिव और मेटल पॉजिटिव. 4 एटम्स आपके इनोलेट के, 2 एटम्स आपके कार्बोनाइल के, तो टोटल कितने बनते हैं? 6. So when 6 groups are or atoms are coming together in 3 dimension form, what will be the appropriate conformation they want to get? 6 member में आपकी कौन सी बेस्ट कॉन्फ़िगरेशन आपने देखी है? साइक्लोहेक्सेन की कॉन्फ़िगरेशन पता है आपको? विच वन इज चेयर, बोट, ट्विस्ट एंड हाफ चेयर. तो उसमें होता क्या है? साइक्लोहेक्सेन इज 6 कार्बन. ओके, सो यू आर सेइंग दैट 6 कार्बन विल बी बेस्ट स्टेबल इन चेयर फॉर्म. उसमें या तो ग्रुप एक्सियल पे होगा या ग्रुप इक्वेटोरियल पे होगा. सो जस्ट इमेजिन आ साइक्लोहेक्सेन मॉलिक्यूल. आप इसको आपस में जब रिएक्ट कर रहे हो, हाउ दे आर गोइंग टू लाइन इन अ स्पेस. नाउ सी इन दिस पिक्चर. फर्स्ट पिक्चर you have carbon oxygen double bond. Forget about A and H. They are either going to take the axial position or equatorial. Now in the enolate form, carbon carbon double bond, oxygen negative and metal. Now how these 6 are coming together in a chair conformation. Now if you see, you have a E enolate. What is the meaning of E? Your R and O negative M, they should be on the opposite side. Now see the picture which I have shown in red color. Carbon oxygen bond is in red color, carbon R bond is in red color. If you have a E enolate, check oxygen and R are opposite to each other. Now if you go to the Z enolate, what is Z enolate? R and O negative M on the same side. तो you see carbon oxygen and carbon are they are group on the same side. So the question comes that with the stereochemistry at carbon 2 and carbon 3 you are able to get preferably either S or either E molecule. So if you are planning a such type of reaction, appropriate enolate is going to give you this. तो अब आप देखो 100% में से आपने 50% तो पॉसिबिलिटी को रिड्यूस कर दिया, या कह सकते हैं 50% पॉसिबिलिटी आपने सेलेक्ट कर ली. ओके, सो फर्स्ट इज द रिलेटिव स्टीरियोकेमिस्ट्री एट कार्बन 2 एंड कार्बन 3. अब मुझे देखो कि आपने 50% ई इनोलेट प्रेफर एंटी एल्डोल प्रोडक्ट, जेड इनोलेट प्रेफर सिन एल्डोल प्रोडक्ट. अब आगे की कहानी 50-50 तो कर लिया आपने, आइदर सिन आएगा और आइदर एंटी आएगा. अब सिन में भी आपके पास 2 है, एंटी में भी 2 है, उसको कैसे सेलेक्ट किया जाए. अब मुझे बताओ फर्स्ट जनरेशन में क्या किया. नाउ आई वांट आंसर फ्रॉम द ऑडियंस. इन फर्स्ट जनरेशन हाउ you how selectivity is done in first generation? You place something on substrate so that the incoming group can be selectively come. Now in this case you have a carbonyl hydrogen and R group, or in your first picture it is ACOH. अब A जो है अगर आप उसको selectively place कर दो तो जो सिन में आपके 2 है वो 1 बन सकता है. कैसे? अब आप इस पिक्चर को ध्यान से देखो. मैंने जो A है, जो आपकी ये पहले वाली पिक्चर में A है, कैन यू सी A? ये जो A है उसको मैंने सेलेक्टिवली इंट्रोड्यूस किया. क्या सेलेक्टिविटी इंट्रोड्यूस की उसमें? हाइड्रोजन इज अबव द प्लेन, मिथाइल इज, और जो आगे O बेंजाइल ग्रुप लगा रखा है वो मैग्नीशियम के साथ चिलेशन भी कर रहा है. नाउ योर इनोलेट इज कमिंग. अब बताओ आपने इनोलेट को कौन सी प्रेफरेंस साइड दी है? द कार्बन नेगेटिव शुड कम टू द कार्बोनाइल कार्बन फ्रॉम द साइड ऑफ हाइड्रोजन और फ्रॉम द साइड ऑफ मिथाइल? From the side of hydrogen, that it is coming from the upper face. Okay, so now from 4 to you move only 2, from 2 to you move only 1. So your enantio selectivity will be the best one in this case. So in any example this is one choice, that you place chirality in your aldehyde. Now if you place chirality on enolate, then you can also select one. So for that you have to prepare one auxiliary, that is your Evans auxiliary. अब आपने इस फाइनल स्ट्रक्चर में देखो, एक तो आपने इनोलेट लिया, इनोलेट कौन सा ले लिया? Z. क्लियर? उसके बाद उसके ऊपर एक कायरल ग्रुप भी फिक्स किया हुआ है. Now can you see 5 membered cyclic ring and there is one group, isopropyl group आपको नजर आ रहा है उसमें? That isopropyl group is lower the plane. Okay, so when you are reacting it, okay, now the reaction in such way that if that isopropyl group is going to create a steric hindrance from the lower phase, the product will be from the upper phase. So in this case I will move to the reaction this. अब आपको चॉइस दे दी, काइंडली सिंथेसाइज नंबर 1 selectively. तो क्या आप करना चाहोगे? You have to selectively synthesize sin enantiomer 1. Okay, so वही 2 लाइन का क्वेश्चन. सबसे पहला क्या करोगे? इनोलेट वो चूज करोगे जो क्या होगा, Z होगा या E होगा? Z, क्योंकि Z इनोलेट ही कौन सा देगा, सिन, E इनोलेट कौन सा देगा, एंटी. So first question, choose Z inolate. Once you choose Z inolate, you will get syn product. अब next step क्या करोगे? Either your aldehyde or either your inolate place a auxiliary which will block the lower phase. चाहे वो आप एल्डिहाइड पे लगा लो. तो जब इस स्टेप में हमने दिखाया ऑक्सिलरी कहाँ प्लेस की हुई है, एल्डिहाइड पे. ओके, जो मीथाइल ग्रुप है उसकी साइड से ऊपर से ग्रुप आ रहा है. आइदर आप ऑक्सिलरी प्लेस कर दो इनोलेट पे, पर ऑक्सिलरी कैसे प्लेस करनी है, लोअर फेस पे आपने कौन सा ग्रुप लगा रखा है, आइसोप्रोपाइल ग्रुप. सो इन दिस वे यू आर able to get only one type of product. So अब बताओ, if you are going to ask asymmetric aldol reaction with enolates, will you able to explain it without even seeing the reaction, without even seeing the mechanism? Okay, this is the general step. Everybody can ask you if you are doing a synthetic work. See, what we are doing in the laboratory, if you are talking about, okay, what we are doing in the laboratory, it is a conventional works. Okay, when you move to the industry, they are making the products which are directly used in the market. Okay, so in that case, whatever the chiral product, if you are in the pharma industry, they are making the drugs molecules where chirality is already associated, because the human biological system, the enzymes, they are stereospecific in nature. They can distinguish the both enantiomers. So that's why they required the pure compound. Okay. So this is a very simple method in 2 line, that the all the 4 products are available. Which type of enantiomers you want to prepare, you can simply tell them by 2-step reaction, that choose appropriate enolate. After choosing the appropriate enolate, either block the phase in your aldehyde, either block the phase in your enolate, and you will get your final product. Okay. So there is another example, that is asymmetric alkylation. Now can you see in your substrate where the alpha carbon is present with respect to carbonyl group? Can you see in your substrate? Okay. So at that position you want to introduce the alkyl group. What is that alkyl group? That is methyl. So in this reaction, if you have to see the product, here you have to introduce the methyl group. For that you are using the methyl chloride as a reagent. Solvent जो use किया है, that is toluene, phenyl methyl, and then aqueous sodium hydroxide. इसमें आपने एक catalyst use किया है, synconine. Okay. Now can you see what is important in synconine? अब इतने बड़े बड़े स्ट्रक्चर्स को आप अगर भूल के एसिमेट्रिक सिंथेसिस और फेस की बात करो, और सारी एसिमेट्रिक सिंथेसिस में एक ही बात रहेगी, जहां आपके पास 2 doors are available, close one, let it be from only one side. So अब आप अपने प्रोडक्ट में देखिए मीथाइल किस साइड से ऐड हुआ है, अबव द प्लेन. सबको विजुलाइज हो रहा है. ओके. कैटलिस्ट में देखिए कायरलिटी कहां है, हाइड्रोजन एंड OH. which one is on upper side? Hydrogen. Okay. So this alkylation of carbonyl is done with the help of this catalyst. ये catalyst कैसे work करेगा? This catalyst have nitrogen positive charge. Okay. it is reacting with aqueous sodium hydroxide moving into the organic, वो पढ़ा है, that phase transfer catalyst. Now this catalyst is moving into the organic phase. Once it is moved into the organic phase, वहाँ पे solvent क्या available है, toluene. Now this catalyst has to direct the reaction. कौन सी generation होगी ये? It is generation 1, 2, 3 or 4? 4. 1 is substrate, 2 is auxiliary, 3 is reagent, 4 is catalyst. Now your catalyst is directing the incoming methyl group to attack the substrate from which phase? From the upper phase, because the chiral unit present in catalyst, you can see hydrogen and OH. So if you see the combination methyl chloride, the methyl is coming, it will come from the side of hydrogen. If forget about this number 2 syncodine, then you have a carbonyl which is planar in nature. Bring the methyl, it can come from up or it can come from below. So that enantioselectivity is done with the help of catalyst. And what is the plus point of catalyst? It can be recovered and reused. So this is the example asymmetric alkylation. Okay. Now this is a very important reaction we have studied, that is the Sharpless epoxidation or asymmetric epoxidation. Now what is the substrate? If I say it is like a allyl alcohol, if you see substrate, you have a carbon-carbon double bond and CH2OH. Carbon-carbon double bond and one carbon, you call it as a allyl unit, and then you have a functional group OH. So that is allyl alcohol. Okay. R1, R2, R3, they are the different group. अब आपने epoxidation करनी है. What is epoxidation? You have to attach the oxygen across carbon-carbon double bond, but you have one query, carbon-carbon double bond is a planar. अब oxygen कैसे जाए? Normal reaction करोगे, it can above the plane and below the plane, but you want to achieve the 100% enantioselectivity. So what you done? You try to put a one phase with lot of steric hindrance with such a bulky group, so that this reaction can occur only from the one side. Now if you see the number 59, what is this 59? This is just the complexation of the reagent which you are using. Which reagent you are using? Titanium tetraisopropoxide plus diethyl tartrate. Diethyl tartrate, you can take Rn, Rr and SS. What is the difference between Rn and SS? This is just the arrangement in the space, whether it has a R configuration with particular carbon or whether it is as a S configuration. So these two are only differentiate with respect to the planes. Okay. So titanium tetraisopropoxide and tartrate, diethyl tartrate, they first come together, make a complex. Okay. Now can you see if we mark it as a 59. Now if you see 59, such a big structure with the substituents like titanium isopropoxide, CO2 ethyl group. So once this is there, now imagine here you can see that this is the sheet and like this. So titanium tetraisopropoxide and diethyl tartrate, they are clubbed together to block one phase. Now what happened? What is your substrate? Allyl alcohol, it is coming to that complex. Okay. Who is going to give oxygen to the allyl alcohol? You use tertiary butyl hydroperoxide. Peroxide means it has a extra oxygen. So it is going to deliver the oxygen. आपने पहले टाइटेनियम और डाइथाइल टार्ट्रेट से एक कॉम्प्लेक्स बनाया, अब उस कॉम्प्लेक्स के ऊपर आप अपने दोनों सबस्ट्रेट को लेकर आ रहे हो. वन सबस्ट्रेट इज योर एलाइल अल्कोहल. इस एलाइल अल्कोहल को ऑक्सीजन देने वाला रिएजेंट क्या है? टर्शरी ब्यूटाइल हाइड्रो पेरोक्साइड. अब आप इसको ध्यान से देखो, that these R groups, they are present below the plane. So your substrate are coming from the above side. Now you can see titanium O alyl group. तो यहां से आपका आ गया tertiary butyl hydroperoxide. Now can you see these red lines? This delivery of oxygen is occurring from the same phase. So in this case you are going to get only one particular type of epoxide. Now if you choose the another tartrate, then alyl alcohol and tertiary hydroperoxide is going to hook from the bottom side, then the oxygen is delivered to the substrate and your reaction is going to occur. So in this reaction the possibility is what? In the complexation stage, carefully check the positioning of the substrate alcohol and the tertiary butyl hydroperoxide, which is going to deliver oxygen through one phase. Check complex 59 where tartrate and titanium form the initial complex. So that is asymmetric epoxidation, where selectively, now in a simple way, without seeing this 59, a simple question asks, what is the difference between epoxidation and asymmetric epoxidation? It is a two-liner question. Forget about 59. You don't know titanium isopropoxide, you don't know diethyl tartrate. What you can say, sir, in carbon-carbon double bond, it is a planar one. If we are going to add a reagent, oxygen can come from the above side, it can deliver from the below side. But what is in sharpless? Sharpless efficiently try to block either the above and below by introducing a complex. What is that complex? He used titanium isopropoxide for the bigger group, and he used for selecting of these two phase either by RR diethyl tartrate or by SS diethyl tartrate. So if you block any of the phase, your substrate are definitely going to go from the opposite side. So if your both substrate are in opposite side, alyl alcohol and your tertiary butyl, you can get only one type of epoxide. So that is the enantiomer selectivity is 100% in this case, only because the closing blocking of one phase by such a 59 complex. Okay, that is the important part of asymmetric alkylation. And I have shown you one very important compound. If you see, that is your L-DOPA. This is a drug which is used for the anti-Parkinson disease. Okay, can you see in this chiral center what you required? One NH2 group, one COH group, and the other side is your hydrogen and aromatic ring substituted by 2 OH. So the industry which is making this drug, okay, they require this drug in pure enantioselective way. What is the difference? Why pure enantioselective way? Anti-Parkinson disease में क्या है? The drug has to reach brain. So brain has a barrier, it has a membrane, it cannot allow every molecule to pass through it. Okay, so out of the two enantiomer, it will allow only the delivery of one enantiomer to that. So that's why its name is L-DOPA, the only one enantiomer. How we differentiate between enantiomer, L or D? So it will only allow one to pass to the brain barrier. Okay, so in this case you have to synthesize this molecule. आपने इस molecule को commercially synthesize करना है industry में. Can you see the substrate? एक कार्बन कार्बन डबल बॉन्ड की अगर आप एल्कीन ले लो तो विच इज इन अ प्लेनर, कैन यू प्ले asymmetric synthesis in that? Asymmetric synthesis can be played on what type of molecule? sp2. sp2 molecules कौन से हैं? कार्बन कार्बन डबल बॉन्ड एंड कार्बन ऑक्सीजन डबल बॉन्ड. Okay, so you chose a substrate which has a carbon carbon double bond. Now can you see in this case your R is this aromatic ring substituted with 2OH. Now यहाँ पे आपके पास एसिड ग्रुप है, यहाँ आपके पास अमीन ग्रुप है. आपने दोनों की प्रोटेक्शन की है क्योंकि आपको फाइनली एसिड भी चाहिए और अमीन भी चाहिए. तो फिर आपने रिएक्शन क्या करना है? ओनली दी हाइड्रोजनेशन. Hydrogenation across carbon carbon double bond. For that hydrogenation you are choosing a one complex. That complex is helping in blocking of the one phase, addition of hydrogen only from the other phase. So the pure enantiomeric product can be achieved by this reaction. Okay, so see what a beauty of this catalyst system कि आपके पास स्टार्टिंग में कार्बन कार्बन डबल बॉन्ड अवेलेबल है, एसिड ग्रुप ऑलरेडी प्रेजेंट है, अमीन ग्रुप ऑलरेडी प्रेजेंट है. उनको जस्ट प्रोटेक्ट कर देना है क्योंकि वो फाइनल स्टेज में हमें चाहिए. दैट इज ओनली प्रोटेक्शन, डी प्रोटेक्शन स्टेप. यू हैव टू जस्ट ऐड दी हाइड्रोजन अक्रॉस दी कार्बन कार्बन डबल बॉन्ड. तो सोचो एंटी पार्किंसन डिजीज के लिए आप जो ड्रग बनाना चाहते हो, फॉर दैट ड्रग व्हाट टाइप ऑफ रिएक्शन यू आर डूइंग? एडिशन अक्रॉस द एल्कीन to get the alkane molecule, which is the first reaction, but you have to do selectively. So for selectively you have chosen that catalyst, and I think you know how the metal which is surrounded by different ligands is acting stereospecifically towards the one phase only. Okay, so the last reaction which we are talking about, the Diels-Alder reaction. In Diels-Alder reaction, what type of group which we are talking? Diene and dienophile. It's a simple step which you are doing. अब मैं आपको ऐसी पिक्चर्स और रिएक्शन दिखाऊंगा जिसमें आप सब्स्टिट्यूएंट्स एंड ग्रुप्स को देखेंगे लेकिन मेन रिएक्शन को नहीं ध्यान में. But what we have to see, whatever the R and Z, ये जो डबल बॉन्ड है ये प्लेन में है. नाउ इमेजिन दिस इज कार्बन कार्बन डबल बॉन्ड, दिस इज योर डाइन, दिस वन एंड दिस वन. ये डबल बॉन्ड हो गया. आप ये बॉन्ड ऐसे बना रहे हो. दिस इज एवरीथिंग इज गोइंग ऑन इन द प्लेन, कोई स्ट्रेस नहीं है. एज यू ट्राई टू प्लेस आर और आर प्राइम और जेड एंड एक्स, वेयर दे आर? They can be either above the plane or below the plane. So in that case you have to study the asymmetric reaction. So if you see in Diels-Alder reaction, a 6-member ring can be easily formed. आपने डाइन एंड डाइनोफाइल को सर्कल में बनाना है, आपके पास 6-member साइक्लोहेक्सीन बन जाएगा. बट कार्बन्स पे जो ग्रुप लगे हुए हैं, चाहे वो X एंड Y है और A एंड B है, ये कैसे हो सकते हैं? तो जब ये आपका सारा सिस्टम प्लेन में है, योर रिएजेंट कैन बी आइदर अप एंड आइदर बिलो. और इफ योर रिएजेंट कैन बी आइदर अप और बिलो, देन व्हाट कम्स? asymmetric synthesis. Then it will enter. Then you can selectively check whether you want to go upside, whether you want to go. इन Diels-Alder रिएक्शन में एक एक्सो प्रोडक्ट बनता है, एक एंडो बनता है. विच वन इज प्रेफरेबली? एंडो. वाई एंडो? because there is a secondary interaction in your endo. So if you see in a reaction that you have a possibility of exo and endo, but secondary interaction के कारण mainly in Diels-Alder reaction you will get endo. If we are talking about endo, now can you replace syn and anti with endo and exo? What is endo product? Can you see here? R and Z either above the plane or below the plane, because they come together, that is endo, which should not be, but due to secondary orbital interaction the stability of endo product is more as compared to exo, and in exo R and Z should be opposite to each other. Now can you see here, first case and second case R and Z are opposite to each other. So if such type of possibility, how we can achieve? We can place a auxiliary. If you see chiral auxiliary blocking the upper face, नाउ आपने ऑक्सिलरी ऐसे प्लेस कर दी है जिसने अपर फेस को बिल्कुल ब्लॉक कर दिया, अब रिएक्शन कहाँ से होगा? लोअर से। अब लोअर में आपका क्या अटैक करेगा? डाइनोफाइल जा रहा है, डाइन तो पड़ी हुई है, अब डाइन में क्या कोई सेलेक्टिविटी अचीव की जा सकती है? इफ आई एम सेइंग इन अ सिंपल वे, ये बहुत ही सिंपल वे में एक्सप्लेन करना है आपने। ये जो मैंने यहाँ पे एक, जब हम Diels-Alder रिएक्शन लिखते हैं, वी राइट इट इन 2 डायमेंशन, कार्बन डबल बॉन्ड, एक बॉन्ड एंड कार्बन। कैन दिस बी, इसमें और इसमें आपको कोई चेंज नजर आया? These 2 double bonds are opposite to each other, or I can write in this way, they are on the same side. Okay, so once आपने एक ऑक्सिलरी प्लेस कर दी, उसने पहले ब्लॉक कर दिया, उसके बाद डाइन भी आपके 2 फॉर्म में अवेलेबल है, वेदर इट इज ऑन दिस सिन और एंटी. नाउ यू ब्रिंग द डाइनोफाइल, यू आर एबल टू गेट only one type of pure enantioselective product. Now in this reaction, don't go about the any other thing, just search in substrate. मैं आपसे just ये last reaction है, I just want to say इस reaction में Diels-Alder reaction possible करने के लिए just select the dine and dinophile. Now can you see in this case, originally the reaction is only between, this is your dinophile, this is your dine. अब जो डाइनोफाइल है, कार्बन कार्बन डबल बॉन्ड, उसके एडजेसेंट में आपने देखो क्या प्लेस किया हुआ है, एक कार्बोनाइल ग्रुप है, देन अ 6 मेंबर रिंग, देन अ फिनाइल रिंग. सो अब आपने इमेजिन ऐसे करना है, ये रहा आपका डाइनोफाइल. ओके, अब इस डाइनोफाइल के नीचे आपने क्या ऐड किया हुआ? कार्बोनाइल ग्रुप, देन 6 मेंबर चेन, देन योर दिस रिंग, बेंजीन रिंग. अब आपकी डाइन कहाँ से इसको एंटर करवा पाओगे? इधर से. तो डाइन कहाँ आ जाएगी? आगे. डाइन यहाँ पे आ गई, डाइनोफाइल यहाँ प्रेजेंट है, दे आर गोइंग टू मेक अ बॉन्ड. सो कैन यू सी हियर दैट वी पिक दिस डाइन, we take it in the front of the dinophile and your reaction is occurring only from the front side. So अगर आप सिंपल बात करेंगे तो फिर तो प्लेन में ये डाइन है, ये डाइनोफाइल है, you are making a reaction. ओके, बट आपने क्या किया, इस डाइनोफाइल को इतना हैविली कर दिया कि डाइन के लिए कहाँ से रहे एंट्री? ओनली फ्रॉम द फ्रंट साइड. अब सिचुएशन क्या आती है जब ऐसा रिएक्शन आता है? हम क्या सोचना शुरू कर देते हैं, ये वाला, ये वाला ग्रुप, बेंजीन रिंग, बेंजोइल और एच, दे आर जस्ट इन द सिस्टम टू ब्लॉक वन फेस. They are not going to participate because what is Diels-Alder reaction? It is only the reaction between diene and dienophile. यहाँ पे मैं कोई भी लगा देता हूँ ग्रुप. This is X, this is Y, this is Z, this is P. ये जिस जिस कार्बन के साथ अटैच्ड है फाइनल प्रोडक्ट में वो वहीं रह जाएगा. ओके. But when we are talking about 3-dimension picture, we can see. Now see in this example also, ये आपकी डाइन है, ये आपका डाइनोफाइल है. आपने क्या इंपॉर्टेंट पॉइंट किया इसमें? कार्बोनाइल के साथ आपने एक कायरल सेंटर लगा रखा है. उस कायरल सेंटर में क्या क्या प्रेजेंट है? हाइड्रोजन, टर्शरी ब्यूटाइल एंड OH ग्रुप. टर्शरी ब्यूटाइल सबसे बड़ा ग्रुप है, वो आपने फ्रंट फेस पे हैंगिंग पे लगा दिया. सो व्हाट यू लेफ्ट अदर साइड यू हैव अ OH और CO. कौन सा मॉडल पढ़ा है हमने? कार्बोनाइल ग्रुप के एडजेसेंट कार्बन में अगर कोई ऐसा एटम हो जिसके पास इलेक्ट्रॉन डेंसिटी हो, देन दे विल क्लब टुगेदर टू मेक अ चीलेशन. So if you are trying to create chelation also, tertiary butyl also, then diene and dienophile will react from which side? From the other side. So the possibility of 50-50 is exactly shift into the other side, that only one type of enantiomer will be there. Okay. Okay. Thank you so much for your attention. Any question? सो इसका मतलब क्या समझूं? आ गया या बिल्कुल नहीं आया? ओके.
We'll resume the lesson in next 5 minutes. Welcome back everyone. We now have Miss Pooja under the supervision of Professor Gurjaspreet Singh, a research scholar, and she'll be presenting a lecture on synthesis and characterization of biologically active triazole compounds.
A very good afternoon everyone. Myself Pooja Malik. Today I am here to present lecture entitled as Synthesis and Characterization of Biologically Active Triazole Compounds. In the first chapter we will study the basic terminology going to use during the lecture: click chemistry, silicon chemistry, silane, silatrane, Schiff base, magnetic nanoparticle and computational studies. Talking about click chemistry, 1,3-dipolar cycloaddition reaction of 1,3-dipolar cycloaddition reaction of azide with alkyne under heating produce an equal mixture of 1,4 and 1,5 disubstituted isomer of 1,2,3-triazole which lack regioselectivity. When copper is used as an catalyst in the same reaction, it has been observed that only 1,4 disubstituted isomer of 1,2,3-triazole is obtained as a single product. Talking about the applications of the triazole moiety, they are used as anti-cancer agents, antimicrobial agents, anti-malarial agent, anti-tuberculosis, anti-leishmania, antiviral, and many more. Silicon. Silicon is one of the most abundant element in the earth crust after oxygen and carbon. The term silicon came from the Latin word silicium, which means hard rock. For the first time in 1824, a scientist, Jacob, identified the silicon element. He synthesized non-crystalline silicon by heating sodium with silicon tetrafluoride. In 1854, Henry prepared the world's first crystalline silicon. Talking about the applications of the silicon, it is used in integrated circuits, biology, solar cells, environment, marine constructions, and silicons. Organosilicons. Nature provides no direct source of organosilicon compounds. Therefore, this branch of chemistry is entirely a product of laboratory research. As shown in the figure, this is the general structure of the organosilicon. It consists of two substances, X and Y, whereas the substance X can be any halide, amine, alkoxy, and Y can be any organofunctional moiety which can be directly attached to the silicon atom or indirectly through a carbon chain. Applications of the organosilicons are following: cross-linking agents, adhesion promoters, surface modifiers, biological applications, nanoscience and nanomaterials. Silatrane. The word silatrane is made up of two words, sila and atrane. The first subpart, sila, refer to the silicon, and atrane refer to the tricyclic ring around the metal atom. Depend upon the metal atom present in the silatrane ring, its nomenclature vary. If the metal atom present is silicon, it called silatrane. If it is phosphorus, it's called phosphatrane. If it's cobalt, cobaltatrane. If boron, boratrane. If the metal atom is silicon but the substance Y differ, then nomenclature is also differ. For example, for oxygen, its name as silatrane. For nitrogen, it's azasilatrane. For carbon, it's carbasilatrane, and for sulfur, thiasilatrane. Applications of the silatranes are in seed germination, sol gel process, organic synthesis, atomic force microscopy, polymer, biological. Schiff base. Schiff base are synthesized by a simple condensation reaction between the carbonyl and amino compounds. They are also known as the nitrogen analogous of aldehyde and ketones, since in these compounds the carbonyl group is replaced by simple amine or azomethionine group. Talking about the applications, they have various industrial purposes as well as broad range of biological activities, which include antibacterial, antifungal, antiviral, antimalarial, anti-inflammatory, anti-cancer, anti-HIV, and many more. Now we will discuss about the azomethane functionalized platform for the selective detection of zirconium ion and its biological property. Talking about the synthesis of the naphthalene 1-amine functionalized Schiff base, 4-hydroxyaldehyde is treated with propyl bromide using K2CO3 as base and DMF as the solvent. The reaction mixture was stirred for 17 hours at the room temperature. After 17 hours, the reaction mixture was added into ice cold water to obtain the final precipitate. This terminal alkyne is treated with primary amine using ethanol as a solvent. The reaction mixture was refluxed for 12 hours to obtain the final product. The characterization of the final product was done using single crystal X-ray crystallography, 1H-NMR C13. In the 1H-NMR, the disappearance of the peak corresponding to 9 to 10 ppm aldehyde group, the peak at the 8.46 corresponds to the new bond of azomethionine moiety CH double bond N, confirm the successful synthesization, whereas similar in the C13 NMR spectrum, the peak at 160 ppm confirms the formation of the CH double bond N bond. In the 1H NMR spectrum, the band at 607 centimeter corresponds to the CH double bond N. Mass spectra also confirm the successful synthesization. Coming toward the application of the synthesized product, it has been observed that when the solution of the synthesized product was treated with different metal ions, no change in the absorption spectra took place. But when zirconium metal is added in the solutions, its absorption band shifted. After successful detection of the zirconium ion, titration was done to find out the limit of detection and association constant. To find out the limit of detection, a plot between absorbance and zirconium concentration is plotted, and formula is used 3 sigma divided by S. The calculated LOD come out to be 31.5 into 10 ki power minus 9 molar, and association constant comes out to be 8 into 10 ki power 4 molar inverse. To find out the stoichiometry between the synthesized compound and the detected metal, job plot was used. It has been found out that the compound and the metal interact with each other in 1:1. After that, to confirm the selectivity and sensitivity toward the zirconium metal ion of the synthesized compound, interference study has been done. As shown in the graph, it has been observed that if in the presence of the other metal ion, the compound can effectively detect zirconium ion, which prove its sensitive and selective detection. Real sample analysis was done to prove the practical applicability of the synthesized compound. For real sample analysis, spike and recovery method was used using 3 different water samples: tap water, distilled water, and drinking water. In all 3 samples, the recovery percentage exceeded 95%, which shows successful detection of the zirconium in the real life. After that, after confirming the stoichiometric ratio between the compound and the metal ion, the metal ligand complex was synthesized using methanol as an solvent. The reaction mixture was stirred at room temperature for 5 hours. After that, metal ligand complex was characterized using 1H NMR and IR spectra. In the 1H NMR, the shift of peak corresponding to the terminal alkyne proton and 2 protons alternate to its confirmed involvement of binding at these sites. IR also confirmed the same binding sites. After that, antioxidant study was done to find out the radical squelching properties of the compound. For this, DPPH is used as the radical source and ascorbic acid is used as the standard to compare the results. Absorption spectra band was plotted for different concentration of the compound with DPPH solutions. It has been observed that as the concentration of the compound increases, the intensity at 517 nanometer band decreases, which showed the successful squelching of the free radicals. To find out the IC50 value, graph between percentage inhibition and concentration for both ascorbic acid and synthesized compound was plotted. As shown in the table, the ascorbic acid give the IC50 value of 0.5 micromolar and compound give 0.693 micromolar, which clearly shows that compound has effective potential antioxidant activity. After that, antibacterial study activity has also been found out for the synthesized compound. Antibacterial activity of the compound was tested against 4 bacterial strain. It has been shown in the table that out of all 3 compounds, compound 182A is the most effective against all 4 bacteria, which shows that its antibacterial activity is better than the other 2 compounds. After that, molecular docking was also done with TLR4 protein to show its effective inhibition by the compounds. Binding energy of minus 8.23 kilocalorie mole inverse show the effective inhibition. Here we will discuss about the design of fluorometric sensor for tin detection using azomethionine triazole organosilane, turn-off mechanism, antioxidant, and molecular docking inside. Talking about the synthesis, The terminal alkyne containing Schiff base was treated with azide using TTHF as a solvent and copper as an catalyst to finally obtain the triazole bearing organosilicon compounds. This characterization was done using 1H-NMR, C13, mass and IR, which clearly shows the successful synthesis. Coming toward its application parts, UV visible analysis was done. It has been observed that when various metal ion was added in the solutions of the synthesized compounds, no metal ion changes its absorption spectra except tin metal ion. After detecting the tin metal ion, its LOD and absorption value has been calculated by plotting the graph between absorbance and tin metal ion concentrations. The calculated limit of detections comes out to be 1.69 into 10 ki power minus 7 molar, and association constant comes out to be 1.22 into 10 ki power 5 molar inverse. After confirming the detection of the tin metal ion by the UV visible study, fluorescence spectral analysis was also done. It has been observed that the samples show high intensity in the fluorescence spectra, and the intensity remain unchanged in the presence of the other metal ion. But in case of the tin metal ions, the intensities decreases sharply, which show the turn-off mechanism by the introduction of the tin metal ion. After that, titration was done to find out the limit of detection and association constant. The table shows the value of the limit of detection and association constant calculated by UV visible and fluorescence, and it can be shown in the table that the limit of detection calculated by the fluorescence spectral analysis is much lower compared to the UV. After that, to find out the stoichiometric ratio between the compound and the detected metal ion, Jobs plot was plotted, which confirmed the 1:1 stoichiometric between the compound and the detected metal ions. To show the sensitive and selective detection of the tin metal ion, even in the presence of other metal ion, interference study was done. And the graph clearly shows that even in the presence of the other metal ions, the synthesized compound is capable of detecting tin metal ions. To prove the practical applicability, real sample analysis was done using 3 water samples: tap water, drinking water, and distilled water. The sample was spiked with 20 microliter in each cases. To show the recovery percentage shows that the tin synthesized compound can be used as a real life sample for detecting tin metal. After confirming the 1:1 stoichiometry between the compound and the tin metal ion, the complex was synthesized. The synthesized complex were characterized using 1NHMR. It has been observed that the 1H proton in the triazole moiety and the protons corresponding to the oxygen atoms were de-shielded in presence of the tin metal ion, which confirm the participation of these binding sites with the metal ions. After that, DFT study was also done to find out the optimized structure and homolumo energy gap. It has been observed that the homolumo energy gap in case of the parent compound is much higher as compared to the metal ligand complex. The lower homolumo energy gap in case of the metal ligand complex shows the extra stability after its interaction with the metal ion. Antioxidant activities was also done for the synthesized compound. For this, the pure DPPH spectra was first recorded, which show an absorption band at 517 nanometer. After that, compound and DPPH solution were mixed in equal volume to find out the change in the absorption spectra. It has been observed that as the concentration of the compound increases, the band at 570 nanometer decreases its intensity, which shows the radical squelching by the synthesized compound. Graph between percentage inhibition and concentration was plotted to find out the IC50 value corresponding to the ascorbic acid and the synthesized compound. As shown in the tables, the IC50 value comparative to the ascorbic acid clearly shows that the compound has an potential to act as an antioxidant compound. Molecular docking was done with E. coli and Ascaris proteins to find out the bacterial inhibition by the synthesized compounds, and binding energy in both the cases in the range of -7 to -8 kilocalorie per mole clearly shows that compound can be used as an antibacterial. In this part, we will discuss about the synthesis of triazole-linked bis-organosilane compounds as dual-function agents, sensing of aluminium ion in aqueous environment, and HIV-1 protease inhibition. Talking about the synthesis of the bis-organosilane compounds, dihaldehyde compound was treated with primary amine using methanol as an solvent. The reaction mixture was refluxed for 5 hours to obtain the bi-Schiff base compounds. Now this compound was treated with propyl bromide using potassium carbonate as an base, DMF as an solvent. The reaction mixture was stirred at the room temperature for 17 hours to finally get the terminal alkynes. This terminal alkyne compound was treated with terminal azide using copper as an catalyst to obtain the triazole bearing bis-organosilane compounds. The final compound was characterized using 1H-NMR, C13-NMR, mass and IR spectra. Talking about its UV visible spectral analysis of other metal ion do not show any significant changes, but in presence of the aluminium metal ions, its absorption, the band at 319 nanometer completely decreases in intensity, which show detection of the aluminium ion by the synthesized compound. After that, titration was done to find out the limit of detection and association constant. Limit of detection was found by plotting a graph between absorbance at 366 nanometer and concentration of aluminium. It has been observed that the calculated limit of detection comes out to be 57.18 into 10 to the power minus 8 molar, and association constant comes out to be 9.89 10 to the power 5 molar inverse. After that, interference study was done to prove the sensitive and selective detection of the aluminium ion even in the presence of the other metal ions. Joke plot was done to find the stoichiometric ratio between the compound and the metal ion. It has been observed that 1:1 stoichiometric ratio was found out. A real sample analysis was done to show the practical applicability of the synthesized compound as aluminium metal ion sensor. Recovery percentage in tap water, distilled water and drinking water exceeded 94%, which clearly shows that the synthesized compound can be used as an aluminium metal ion sensor in real life applications. Proposed binding mode of compound with the aluminium has been done using 1H-NMR. The synthesized complex was characterized using 1H-NMR. The changes in the peaks in the 1H-NMR corresponds to the proton in the triazole ring, and CH2 directly attached to the triazole ring shows that they are involved in the binding with the metal ions. After that, density functional theory was also done to find out the homolumo energy gap and optimize structure in case of the compound and the metal ligand complex. The homolumo energy gap in case of the compound and metal complex has been observed less compared to the parent compound, which show the extra stability after its binding with the metal ion. Antioxidant activity was also done for the synthesized compound. It has been observed that as the concentration of the compound increases, the band at the 570 nanometer decreases in its intensity, which shows the successful squelching of the free radical present in the solutions. Same experiment was done for the ascorbic acid, and percentage inhibition versus concentration graph was plotted to find out the IC50 value. The IC50 value of the synthesized compound clearly shows that it can be used as an antioxidant. Molecular docking was done against HIV protein, and to compare the result of the synthesized compound, the same protein is also docked with the local available HIV drug. The more negative value in case of the synthesized compound shows that it has potential to inhibit the HIV. Conclusions. That chapter underscored the successful synthesis and characterization of bis 1,2,3 triazole linked bisorganosilane. It particularly emphasized its efficiency as a sensor for aluminium with exceptionally low detection of limit in water methanol solvent system. Its practical applicability was validated through successfully detection of aluminium in real water sample with recovery percentage of more than 94%. Additionally, bisorganosilane exhibit additional radical squelching activity, highlighting its potential as an antioxidant. Furthermore, molecular docking study revealed its promising inhibitory activity against HI1 protease with a binding energy of minus 9.27 kilocalorie mole inverse, which is again compared with the AZT drug which is common available HIV drug. Thank you. सर इसमें हम प्रैक्टिकली चेक करते हैं, हम लोएस्ट कंसंट्रेशन से शुरू करते हैं 1 से 2 से 3 माइक्रोमोलर से, कि किस कंसंट्रेशन पे वो डिटेक्ट कर पा रहा है, वो सेम बैंड देगा जो नॉर्मल मेटल्स के उसने दिया था। जब वो सेम बैंड अपीयर हो जाता है तो हम उस कंसंट्रेशन पे रिकवरी फाइंड आउट करते हैं। ईजी हो जाती है सर कैलकुलेशंस। Sir, using the formula absorption obtained in the using the compounds, and then it's pure sample.
Any justification why it is for aluminium 3? Why not any other trivalent metal ion? Why only for the aluminium 3? Why not any other trivalent metal ion?
So the compound only detected the aluminium metal ion rest.
But any specific reason on the basis of HSA principles or something?
सर उस पर डिपेंड करता है, चार्ज अपॉन साइज रेशोज पे वो मेटल के साथ मैच हो रहा है या नहीं, मेटल के लोन पेयर या उसके इलेक्ट्रॉन्स कितने हैं, बाइंडिंग साइट्स कितनी अवेलेबल है हमारे कंपाउंड्स पे है। यस, कैविटी।
That was a very insightful lecture. Next up, we have Miss Poonam, and she'll be presenting the lecture on platinum complexes in therapeutics.
My topic is platinum complex in therapeutics. फुल स्लाइड करना। अच्छा ठीक है, नहीं कोई नहीं, ठीक है। Metal-based drugs and their role in medicine. Some essential metals that our body need are sodium, potassium, magnesium, calcium, iron, zinc, copper, chromium, manganese, cobalt, etc. and its biological role for enzyme activity, oxygen transport and also growth and metabolism of our body. Discuss the role of the sodium and potassium in biological system. Sodium and potassium are very two important alkali metal and play a very visual role in biological system. These are highly mobile positive cations and forming the soluble salts. They are strongly solvated in the waters. Some foods are, that is tea, coffee and nuts, etc. Sodium deficiency. Due to sodium deficiency, our body excessive sweating, kidney adrenal disorders, and drinking of the too much waters. Some symptom of the adrenal disorders, that is the hypotension, hypoglycemia, hair loss, back pain, confusion and the psychosis, fever, loss of the consciousness, vomiting, joint pain. Now, sodium metal used in the drugs, that is the diclofenac sodium. It is used for the arthritis pain reliefs, and the sodium bicarbonate. It is a oral powder and used for the antacids, and the naproxen sodium used for the headache pain and also the pain reliever. Iron deficiency, that is the anemia. Anemia results from the insufficient oxygen supply due to the decrease the hemoglobin level in our body. It's about 65 to 70% of the total body iron is found in the hemoglobins. Some countries like USA, food especially flour based, are enriched with iron to prevent the anemia. Some iron-rich food for vegetarians, that is the cooked spinach, dried fruits, Brussels sprouts, leafy greens, peas, nuts, broccoli, etc. Medicine role of the iron tablet like ferrous gluconate and the ferrous sulfate with folic acid and some injectable iron, that is the ferric carboxymaltose, iron isomaltosides, ferric derisomaltose and the iron sucrose, and mostly folic acid used for the pregnant woman. Zinc in the human bodies, how much the zinc present in our body. Some discuss are skeleton muscles. In skeleton muscles, that is the 60%, and in a bones, that is present in the 30%, and some small amount present in the skin, that is the 5%, and also the other tissues like brain, kidney, pancreas, hearts, that is 5%. Some zinc amount present in plasma, that is the 0.1%. Most common reason for zinc deficiency, that is the leaky gut and the digestive disorders, and also the medication use, which medicine like acid blockers and the antibiotics, which reduce the deficiency in the our bodies, and also the poor diet and the blood sugar unbalance in our bodies, and chronic stress and high toxic exposure, which causes the zinc deficiency in our body. Now, zinc deficiency symptoms like growth retardation, loss of the appetite, impaired immune function, infection, hair loss, diarrhea, eye and the skin lesions, weight loss, delay wound healing, taste abnormalities, cognitive impairment, mental lethargy, and also the pregnancy complications. Use of the zinc in a medicines. Firstly, zinc lozenges. It is a it is a drugs used for the cold relief, and the zinc oxide ointment. It is a cream used for the skin treatment, skin protections, and also used for the cuts, and the healing relief. And the zinc sulfate. It is a liquid form of the zinc used for the children's. Introduction to the bismuth compound. Bismuth is a heavy metal with a low toxicity, and its common oxidation state +3, and also form the insoluble salt useful in medicine. Bismuth subsalicylate is the most widely used compound in the drugs form. Medicinal use of the bismuth subsalicylate and the pepto-bismol, these two drugs used for the quickly relief in the stomach pain. Why the magnesium is important for health? It is the cofactor in over 300 enzymatic reactions, also regulate the nerves and the muscles functions of our body, and support the energy productions, and essential for DNA and RNA synthesis, and also maintain a normal heart rhythm of our bodies, and also the balance the blood sugar of our body and insulin regulation. Dietary source of the magnesium, that is the banana, dark chocolate, nuts, etc. Magnesium based drugs, magnesium sulfate, it is used for treatment of the seizures in the pregnancy, and magnesium hydroxide used for the antacids, and magnesium citrate used in the bowel cleansing before the surgery, and magnesium oxide drugs used for the dietary supplement and also the antacid. Hypocalcemia, it is due to the deficiency of the calcium in our bodies, that is the hypocalcemia. Vitamin D deficiency reduce the calcium absorption, hormonal imbalance, that is the low thyroid hormone, kidney diseases or malabsorption disorders. Symptom calcium deficiency, that is the weak or soft of the teeth, and also the not eat well by the baby, and the white patches, so the white patches on the baby skin, which is the causes of the deficiency of the calcium in our body. Metal as a carcinogenic. Firstly, nickel. It is found in alloys and batteries, and also due to this, lungs and the nasal cancers. Chromium metal used in paints, dye, and the steels causes of the lungs cancer. Cadmium metal used in batteries, plastic, smoking, and it causes the prostate, kidney cancers. Cisplatinum. Cisplatin a milestone in metal-based chemotherapy. Cisplatinum, it is the first metal-based drug approved for the cancer treatment. Discovered to be highly effective in mice, but later translate to the human used, and also the treat various solid tumors, that is the testicular, ovarian, bladder, head, and the neck cancers. Metallocene halide as a potential anti-tumor agents. Examples are titanium halide, metallocene, and the ferrocenes. It shows the activity against experimental tumors in animals, and also the higher tolerated doses up to the 200 milligram per kg than cisplatin. Cisplatin, it favor the toxic side effect despite the higher dose, and but its delay use, clinical use due to the failure in the mouse leukemia screens. Platinum based anti-cancer drugs. Platinum containing the platinum metal containing medication, that is the cisplatinum, when the drugs stuck to the DNA and kills the cancer cells. History of the discovery of the cisplatinum by the Dr. Barnett Rosenberg in the 1960. A platinum compound from electrode reacted with the buffer solution of the ammonium chloride, inducing the filamentation. This filamentation linked to anti-tumor activity, and cisplatin firstly tested in mice with sarcoma 180 tumors, that is leading to tumor shrinkage and the cure. At last, FDA approved the cisplatin in 1979 as a leading anti-cancer drugs, and also its cisplatin important in inorganic stereochemistry. There are the 5 drug development phases. First is discovery and development of the drug, and second preclinical research of the drug, third clinical research of the drug, and FDA review. At last, Food Drug Administration post marketing safely monitoring the drug for safety of the drug. Type of the cross link. Firstly, A, cisplatin and the carboplatin, both are the platinum metal containing drugs used for the anti-cancers. Chemical binding mechanism. Firstly, initially binding, the first and rate determining step in cis-DTP binding to the DNA is chloride loss and forming a monoaqueous complex that's bind to the DNA via nitrogen 7th of the purines, guanine, and the adenine. Secondly, adduct formation. Platinum form the monofunctional and then bifunctional adduct, stabilize the DNA structure and change the pK value of guanine, indicate the electronic perturbations. And third, ligand retentions. The ammonia ligand on a cis-DTP remain bound to platinum during DNA interactions. This has been verified via radio labeling and the immunological methods. And the stability of the platinum DNA adduct are kinetically inert, but it's stable against physical and enzymatic degradations, but can be displaced by the cyanide or thiourea. Physical change to DNA. When the cisplatinum bind to the DNA and it take place the physical change to DNA, firstly it cis-DTP induce a 34-degree bend into the major groove of DNA via intrastrand cross link, and also unwinding each cis-DTP adduct causes the 13-degree unwinding of the DNA helix. Due to the binding and unwinding, DNA become the shorted and the more mobiles, as shown in supercoiling and the nickel DNA experiment. Cisplatin testing on the cancer cells. We discussed the two cancer cells, HeLa, that is the cervical cancer cells, and L1210, it is the leukemia cancer cells. HeLa cells are a human cervical cancer line famous for being the first human cell to survive indefinitely in a laboratory culture. L1210 cell, on the other hand, are a mouse leukemia cell line used in the cancer research, particular for studying the chemotherapeutics agents and the drug resistance. Graphical evaluation of the cisplatin toxicity, that is cis-DTP, cause a steep drop in cell survival with minimal DNA platinations, but trans-DTP show a gradual toxicity needing higher DNA binding for the similar effects, but also its high potency at a low dose makes the cis-DTP a promising drug candidate despite lack of the perfect screening method. Understanding how a drug works, firstly find out the which part of the cell that drug target, study the chemical change the drug may undergo in the body fluid, and find and study of the drug target adducts. At last measure how stable the adduct are and how long they last. Mechanism knowledge to improve the drug. Firstly, molecular biological tools to monitor the change in a DNA or proteins. Second, genetic method to study how cells respond to treatment of the cancer cells. Immunology approach to detect the cell stress and immune response. At last, the link mechanism with clinical structure activity relationship for better drug design. Clinical picture and the chemotherapy. The most effective against tumors, especially testicular cancer, when the combination chemotherapy with the vinblastine and the bleomycin improve our outcomes against the cancer cells. Response also seen in the ovary, head and the neck, lungs and the cervical cancers. And cisplatinum, when cisplatinum combined with radiation therapy enhance the efficacy. But side effect of the cisplatin are kidney toxicity was a major concern mitigated by hydration and the dieterates. Other side effect also nausea, vomiting, bone marrow suppressions, neurotoxicity, allergic reactions are also take place. Carboplatinum is safer derivatives. It is the bidentate dicarboxylate ligand replacing the chloride, but bulky ring, due to the bulky ring reduce the reactivity compared to cisplatinum. Its advantage of the carboplatinum are reduce the nephrotoxicity and the gastrointestinal side effect. It is the more stable drugs and easier dosing and the fewer the hospitalization. Other generation, next generation is oxoplatin. It is 1,2-diaminocyclohexane. It active even in some cisplatin resident tumors, and but it less nephrotoxicity but may cause of the neuropathway. Cis versus trans DTP DNA targeting firstly unbound cis and the cis and DNA, cisplatin and the DNA, and the B chloride ions are moved and the complex are formed between the cisplatin and the DNA bases, and C 1,2 intrastrand cross-link complex form between the DNA and the cisplatinum. But in the transplatin DNA, D shows the unbound transplatin and the DNA, and chloride ion are moved and form the transplatinum complex with the DNA in a F long range intra or interstrand cross link formed with the DNA, and last rearrangement of the interstrand and the long range interstrand form the targeting cisplatinum and the transplatinum DTP. Cisplatinum versus transplatinum, DNA bind and the adduct formation. Cisplatinum form the intrastrand cross link that disrupt DNA replication, but transplatin form the less effective adduct that are more easily repaired. Secondly, sequence specifically, cisplatin drugs target the guanine sequence, but transplatinum prefer the guanine and other bases of the DNA. And third, DNA structural effect. Cisplatinum bend the DNA toward the major group, adding protein recognitions, but transplatinum causes a hang-like band less recognized by repair machinery. Platinum DNA adduct reductions, antibody-based reductions specific for cis-platinum adduct distinguish between active and inactive platinum drugs. Its clinical relevance: antibodies recognize the adduct in tumor-bearing mice. It detects the cis-platinum DNA adduct in the white blood cell of the patients. Monoclonal antibody studies confirm the adenine-guanine and the guanine-guanine intra-1 to intra-strand cross links and recognize the cis-DTP and the carboplatinum induce the DNA change, but trans-platin DTP adduct the guanine and other guanine based also identify where N is guanine cytosine and the thymine adenine base. Cisplatin DNA binding by the NMR. Cisplatin bind to the at the 7th position of the nitrogen atom of the guanine in the guanine-guanine sequence. Binding the causes a chemical shift in the at the 8th position of the proton and the lower the chemical shift value of the nitrogen at a first position by the 2 units. These shift confirm the nitrogen at a 7th position coordination site for platinum. Thermal effect of the cisplatin on the DNA. As the temperature increases, those immuno signals get broader and meaning that DNA is starting to melt. The melting seems to start away from the platinum sites. It's suggesting the cross-link make the area more stable, and temperature-dependent NMR study used to monitor the DNA stability. Immunoproteins are visible at a low temperature. Chemical and biochemical characterizations. Its restrictions enzyme normally cut the DNA at a specific sequence, but it cannot cut the DNA. This means cisplatin has formed an adduct at the spot blocking the enzymes. DNA polymerase assays platinum adduct inhibit the replications of the cancer cells and its effective block the scene for the guanine and the adenine platinum adduct and demonstrate the replication arrest by the most common cisplatin adduct. Hydrolysis reaction of the cis-DTP chloride ion replaced by the water and follows by the deprotonation and water exchange steps. Its multiple equilibria depending on the pH and chloride concentration, but it's verified by the 195 platinum and the 15 nitrogen NMR spectroscopy. 195 platinum and the 15 nitrogen NMR in cis-platinum studies where platinum bind to the nitrogen or oxygen on a DNA and 15 nitrogen label ligand improve the visibility of the bond strength and the type of nitrogen involved in the cis-platinum drugs. Chemistry of the cis-DTP in biological media. Cis-ligand substitution follows the rate law that it is the second order reactions at a low ligand concentration. Solvent assisted pathway dominate and the hydrolysis of the chloride ligands activate the cisplatin for the DNA binding. Role of the stereochemistry. Only cis-DTP are active, but trans-DTP is inactive despite DNA binding. Indicate that geometric arrangement is key to the biological function. It suggests that DNA binding mode and structural distribution are the crucial. Supporting evidence for DNA targeting. Platinum DNA adduct responsible for the cytotoxicity and great sensitivity in DNA repair deficient cells. Most platinum in cell found bound to the DNA, but platinum DNA binding inhibit the replication and stop the tumor growth of the cancer cells in our body. Functional state of DNA and binding. DNA function influence the platinum binding. It's possible prefer for activity transcribed DNA or single strand regions analogs to functional alkylators preferring the open DNA regions. Worth exploring whether the cis-DTP bind the transcriptions for open combination. Is platinum unique? Unique. Platinum is effective but not necessarily unique. Geometrical matters. Example called octahedral platinum in the 4th oxidation state have the steric classes. So, platinum 4th drugs like tetraplatinum are prodrugs reduced in viva to the platinum 2nd oxidation state. Non-platinum anti-tumor complex are also used for the anti-tumors, that is the palladium, rhodium, rubidium. So the promise platinum react faster may increase the toxicity, and also platinum in the second oxidation state form the chelation or the organometallic approach can manage the reactivity. Collaborations with biologists essential for screening and the biochemical testing. Conclusions: metals ions are vital in the biology and the medicine. Cisplatinum launched the metal based cancer therapy. Safer drugs like carboplatinum followed. New metals, rubidium, rhodium, show the promise, and futures are targeting the less toxic metal based treatments. Thank you.
Any quick question? Anyone have a question? No question. Okay, thank you. ठीक है भाई, कल मॉर्निंग लेकिन 9:15 पहुंचना है क्योंकि प्रोफेसर संजय वर्मा का जा सकते हो, ठीक है, थैंक यू।
Thank you everyone for participating. Have a wonderful evening and we'll see you tomorrow for the final day of the course.